Welcome to MedicoMedics. This is our complete microbiology and infectious diseases lecture series. Over 3.5 hours and 14 chapters of high-yield content, covering everything from the basics to advanced topics. If you find it helpful, please like, comment, and subscribe. It really helps us grow and lets us know what to cover next. So let's get started. So in this almost 4-hour long lecture series, we will begin with Introduction to Microbiology. We will then continue with discussing bacterial cell structure and function, microbial genetics, deep dive into virology, mycology, and parasitology. We then turn our attention to the basics of immunology, discuss host-microbe interactions, principles of sterilization and disinfection. We then move on to antimicrobial agents, discuss clinical microbiology, laboratory and epidemiology, emerging and re-emerging infectious diseases, and end with zoonotic diseases. So as you can see, it's a pretty comprehensive and complete lecture series. So let's begin with Chapter 1, Introduction to Microbiology. So in this lecture, we will discuss what is microbiology, its history, scope, and branches. We will talk about classification of microorganisms, talk about different techniques used in microbiology. Further on, we will continue talking about infectious diseases, antimicrobials, and vaccines. Mention the future of microbiology and end with a summary. Now, what is microbiology? So the definition is the study of microorganisms that are too small to be seen with the naked eye. And it includes bacteria, viruses, fungi, protozoa, algae, and prions. Now, importance. Well, it is super important to study these microorganisms to understand infectious diseases. Furthermore, it has a lot of application in biotech, environment, and public health. For example, E. coli helps digestion, but can cause food poisoning. So by studying this microorganism, we will understand that A, it actually does aid in digestion, but B, it can cause food poisoning. Now, we mentioned bacteria, viruses, fungi, protozoa, etc. So here we have some examples. Here we have the protozoa type, ectoparasite, helminths, and an illustration of a virus. As we move on in this lecture series, we will talk about each of these in detail. So microbiology is like peeking into a hidden world of tiny creatures. These organisms, indeed too small to see with our eyes, play massive roles. Like we mentioned, for example, E. coli helps us digest food, while others may cause diseases. Now, some brief history of microbiology. So we have some early observations made. We have Antoine van Leeuwenhoek. I have no idea if that's how you pronounce that, but let's go with that. Already in 1964, he observed for the first time animalcules under a microscope. And moving on, in 1861, Louis Pasteur did these experiments where he disproved spontaneous emergence. I'm not even going to pretend to know what that means, but, you know. So for a long time, people didn't know that tiny invisible organisms actually existed. And these kind of scientists, like Pasteur and Antoine, discovered this hidden world and proved that these creatures actually could cause diseases. And this was the start of fighting diseases scientifically. Now, we have a bacterial theory of disease. So Robert Koch demonstrated specific microbes causing specific diseases, known as his postulates. And then we had the discovery of antibiotics, famously Alexander Fleming in 1928 discovered penicillin, the first antibiotic. Now let's briefly mention the scope and branches of microbiology. So we have clinical microbiology, involved in diagnosis of infections and testing for antimicrobial susceptibility. We have environmental microbiology, role in the nutrient cycle. We have industrial microbiology, so microbes in the production of insulin, beer, biofuels, etc. We have medical microbiology, so the study of pathogens and their interaction with the host. And this is what we will mainly be focusing in since we're studying medicine. Now, some techniques in microbiology. So we have the microscope, we have light microscopy. So we have do an observation of stained bacterial cells. We have electron microscopy, detailed structures of viruses. So example could be light microscopy, could be used to display E. coli during gram staining. Now, normally we think about microorganisms as these invasive pathogens entering our body, causing harm. But we forget that actually we have a lot of microorganisms normally in our body and on our body. So for example, we have this skin microbiota that helps prevent colonization by pathogens. So it actually protects us. Another one is found in the gut, which helps us with digestion. Furthermore, maybe you've heard of lactobacillus, another microorganism that helps maintain vaginal pH. So our body is actually home to a lot of friendly microbes that protect us and keep us healthy. However, if these quote-unquote good bacterias get out of balance for whatever reason, other harmful microbes might take over and then lead to pathology. Now, moving ahead from what is normal to infectious diseases caused by microorganisms. So we mention microbes as pathogens, where we say pathogens versus opportunistic pathogens. Now, pathogens are microorganisms that cause disease, while opportunistic pathogens cause illness primarily in individuals with weakened immune systems. Now, some examples of infectious diseases: bacteria, so tuberculosis or Mycobacterium tuberculosis, which is actually a serious respiratory disease that primarily affects the lungs. Another one is caused by viruses, like influenza, which is a contagious respiratory illness caused by influenza viruses, often leading to seasonal outbreaks. Then we have fungi. So candidiasis, an infection caused by Candida species, commonly affecting the skin, the mouth, and genital areas. So this differentiation highlights the diverse mechanisms through which pathogens affect human health. So we've mentioned now the general types of microorganisms and pathogens like bacteria, protozoa, virus, etc. We then moved on to talk about normal microbiota. So the presence of microorganisms in our body and on our body, for example, on our skin. And these have protective features. But we then moved on to pathogens and opportunistic pathogens, meaning infections caused by microorganisms. Now, to combat these diseases caused by, for example, viruses or fungi or bacteria, we have antimicrobials. For example, we have antibiotics. And antibiotics work by targeting bacterial mechanisms. For example, cell wall synthesis. So we use penicillin to inhibit the synthesis of bacterial cell wall, essentially the formation of the bacterial cell wall, which leads to bacterial death. Against viruses, we use antiviral drugs, and these interfere mainly with virus replication processes, reducing disease severity. So it doesn't eliminate it, but it reduces its severity. Another approach we have is using vaccines. We have live vaccines like the MMR vaccine, and MMR here stands for measles, mumps, and rubella. And these live vaccines contain weakened pathogens to stimulate immunity without causing disease. So you take a little bit of this pathogen, let's say, and you put it in the body, enough to stimulate our antibody production. So the immune system produces a protective mechanism towards this pathogen. And then that immune cell begins to replicate, producing a lot of them. And remembering the characteristics for this specific pathogen. So we had enough of it to stimulate this immune response in our adaptive immune system, but not enough to create illness. So in the future, when this pathogen presents itself, we already have a massive defense system ready to attack it. We also have inactivated vaccines, whereby we use killed pathogens or parts of the microbe to, again, trigger an immune response. For example, the polio vaccine. So in summary then, microbiology underpins our understanding of health, disease, and the environment. We discussed the role of microorganisms in the human body and ecosystem. We talked about microorganisms. We mentioned normal microbiota, pathogens, opportunistic pathogens, etc. Furthermore, we have techniques such as microscopy and culture in diagnostics. We can also add the use of vaccines, the use of antibiotics, antivirals, etc. in treating these different infections caused by these microorganisms. And that's the end of this first lecture. Continue now to Chapter 2. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 2, Bacterial Cell Structure and Function. In this extensive lecture, we will talk about bacterial cells, give you an overview of bacterial morphology, talk about the bacterial cell envelope, the plasma membrane, the concept of the cell wall, Gram staining, and further continue on talking about cytoplasm, endospores, mechanisms of transport, cell division, etc. And then we will end with a recap and conclusion. Now, bacteria are single-celled prokaryotic organisms, and they have unique features like they lack membrane-bound organelles. They possess specialized structures for survival and adaptation. Why is this important? Because understanding their structure helps explain their function, their pathogenicity, and antimicrobial targets. So developing antibiotics, for example. Now, overview of bacterial morphology. So let's talk about some shapes and arrangements. We have cocci. These are spherical. For example, the bacteria Staphylococcus aureus. And here is an illustration. We have bacilli. These are rod-shaped. You can see that here. For example, E. coli. We have spirilla. These are spiral-shaped, like Treponema pallidum, seen here. And Treponema pallidum, if you didn't know, is the one causing syphilis. Then we have Vibrio. This is comma-shaped and causes Vibrio cholera. It looks like that. Then we have pleomorphic bacteria, and these have a variability in shape, for example, Mycoplasma. Now, cellular arrangements. So we can have chains, like for example, Streptococcus. These can be in clusters, like Staphylococcus, and they can be in pairs, like Diplococcus. So all in all, bacteria come in different shapes, sizes, and forms. Here then we have an illustration of these cellular arrangements. So as we mentioned, the chains seen in, for example, Streptococcus, we can see here the pairs in Diplococci. You can see it here. And of course, clusters in Staphylococcus, you can see illustrated here. Now let's continue on to bacterial cell envelopes. Now let's talk about the bacterial cell envelope. So what are the layers of this cell envelope? We have the plasma membrane, seen here. We have the actual cell wall, seen here. And then a capsule or a slime layer, if present. Now, what is the function of the cell envelope? Well, to protect, shape, and for selective permeability. So the cell envelope is like a bacteria's armor. It's protecting it from the environment. And as mentioned, it has multiple layers. And it has a membrane which controls for what goes in and out. And this envelope, what you need to remember is that this cell envelope is what makes bacteria tough and resilient. Now let's talk about the plasma membrane. So the plasma membrane is like the security gate of the bacterial cell. And it controls what nutrients and waste enter or leave. And it also helps the cell to generate energy. Now, some antibiotics work by damaging this gate, killing the bacteria. Now, its structure, it's a phospholipid bilayer with embedded proteins. Its functions, selective permeability, so transport of nutrients and waste. Furthermore, as we mentioned, the electron transport chain is here for energy production. And as we mentioned, why is this clinically relevant? Because it is the target of, for example, polymyxin antibiotics. Now, as we mentioned before, we said that the bacteria has this cell wall, and we differentiated between gram-positive and gram-negative, where gram-positive has a much thicker cell wall compared to gram-negative that has a much thinner cell wall. Now, the composition for the cell wall, whether it's positive or negative, is that it consists of peptidoglycan, also known as murin. And this peptidoglycan is a polymer made of two repeating sugar molecules that form a glycan backbone. Okay, so we will find sugar here, derivatives of glucose, essentially. And this forms a glycan backbone. Now, these sugars then are linked to short peptide chains. All right? Short peptide chains. And it's these structures that make it both strong and flexible, allowing the cell wall to resist, for example, osmotic pressure, while still being somewhat adaptable. So when we talk about peptidoglycan or murin, We have here the N-acetylglucosamine, NAG, and acetylmuramic acid, NAM. Now, NAG is a derivative of glucose. Indeed, the NAG is a derivative of glucose and is one of those repeating sugar units in the backbone of peptidoglycan, while NAM, or acetylmuramic acid, this is a modified form of glucose. And what differentiates it is that it has a lactic acid group attached to it. So it is the other sugar in the repeating backbone. Remember, we said that peptidoglycan, or murin, is a polymer made of two repeating sugar molecules, and these form the glycan backbone. So it's these two. And then we had these peptide chains cross-linking them. And here in the illustration, you can somewhat see. This is the peptidoglycan, right? Thick wall, because we're talking about gram-positive. All you need to remember right now is that it has a thick peptidoglycan layer. But you can see these cross-links here. Now, just to mention another thing, these peptide chains, right? These peptide chains are cross-linked to each other in this peptidoglycan mesh. And this cross-linking indeed is what provides additional strength and stability to the entire structure. Now, we have gram-positive. Indeed, it has a thick peptidoglycan layer. We also have teichoic and lipoteichoic acid here, as you can see illustrated here. While in gram-negative, we have a thin peptidoglycan layer. But we also have what differentiates it from the positive, gram-positive bacteria is that it has an outer membrane with lipopolysaccharides, LPS. We will look at this in the next slide. Now let's put the gram-negative and gram-positive in comparison and dive into detail to differentiate between them. Hopefully, after this slide, you will have a list and a way to identify the differences between them. So we mentioned that starting with the cell wall structure, okay? Now, the gram-positive bacteria had a thicker or a thick peptidoglycan layer. You compare that to gram-negative, it is much thinner, as illustrated here. Furthermore, this peptidoglycan layer that is thick in gram-positive provides rigidity and strength to the cell wall. But another thing that differentiates the two is that gram-positive does not have an outer membrane. If you look at gram-negative, you find an outer membrane here. And this outer membrane, which is outside, right? It's outside the peptidoglycan layer that the gram-negative bacteria has, also then acts as an additional barrier. So that's one main difference, or two main differences. Then we have gram staining procedures, right? Where we want to identify when a cell is either gram-negative or gram-positive, or vice versa. And when we do a gram stain on these bacteria, if it's gram-positive, you will have purple or violet after gram staining. So it will turn purple or violet. And this is happening due to the thick peptidoglycan layer which retains crystal violet stain. We use crystal violet as part of gram staining procedure. This is not something you need to learn or understand immediately now. We will briefly mention it as well in the next slide. But essentially, we apply crystal violet and iodine and alcohol and some other things, and the stain will then turn a certain color based on if it has a thick layer or a small layer. So it's worth repeating. This is high yield. This is asked in a lot of medical tests. So gram positive, purple or violet. And why is it turning purple or violet? Because of the thick layer retaining this crystal violet that we are using. Now in gram negative, it's going to turn either pink or reddish. And this is indeed due to the peptinoglycan layer. Because it's thin, it cannot retain the crystal violet stain. And instead, it's also part of the procedure, safranin, we call it the counter stain, that will color the bacteria instead, and it will turn pink or reddish. So pink or reddish, gram negative, purple or violet, gram positive. There we go. So that's two main things. The cell wall structure is different, right? One has an outer membrane, the other one doesn't. One has a thick peptidoglycan layer, the other one doesn't. One turns purple during staining, the other one doesn't. Third, we mentioned already briefly the outer membrane. So in gram positive, this outer membrane is absent. In gram negative, it's present. It's a double-layered outer membrane containing lipopolysaccharides. And this is contained on the outer leaflet, as you can see here. And it can actually trigger immune responses. So let's briefly check into this lipopolysaccharide that we have identified existing on the outer part of the outer membrane of the gram-negative bacteria. So lipopolysaccharides, first of all, are absent in the gram-positive bacteria, but in the gram-negative, they're present. And the lipopolysaccharide, particularly the lipid A portion of it, is a very potent endotoxin, and that can cause severe immune reactions in humans. And if you say, hey, it's unfair that the gram-negative has all of these things and the gram-positive doesn't, well, the gram-positive has something else. It has these teichoic acids, and these are embedded inside of this thick peptidoglycan layer. And these acids help with the cell wall maintenance and contribute to the negative charge of the cell. And the gram-negative don't have that. Instead, they have these lipoproteins in the outer membrane. Another difference is the periplasmic space, seen here, right? And in gram-positive, it's very narrow or even absent. And there is only a small space between the plasma membrane and the thick peptidoglycan layer, as you can see illustrated here. This layer, this space here is very, 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 very small. While in gram-negative, right, in gram-negative bacteria, it is present, and the periplasmic space is very well defined and located between the inner membrane and the outer membrane. and it contains enzymes and transport proteins, etc. Now let's turn a bit more to the clinical part of all of it. So what is the difference in sensitivity, for example, to antibiotics? So gram-positive bacteria are more sensitive to antibiotics like penicillin. Why? Because penicillin targets peptidoglycan synthesis. So it is more easily targeted because their thick peptidoglycan layer is more exposed, right? It's bigger, it's more exposed. And gram-negative is less sensitive to many antibiotics due to the outer membrane that it has, right? It has an extra outer membrane here acting as a barrier. So it makes it harder for antibiotics to penetrate and reach this peptidoglycan to cause its harm, or in another way, to heal us. What about resistance to phagocytosis? Well, gram-positive can be susceptible to phagocytosis, although the thick peptidoglycan layer sometimes make it harder for immune cells to actually engulf it. And the gram-negative ones, especially the lipopolysaccharides that are here expressed on the outer surface of the outer membrane, they can act as a shield against our immune system. So it makes them more resistant to phagocytosis. And if you don't remember, phagocytosis, essentially you have these cells, macrophages, scouting around for pathogens or something that should not be in our body. And if it detects it, it will try to eat it up, engulf it, and break it down inside and then spit it out. So that's the context here. Now, let's see an example here. We have Staphylococcus aureus or Streptococcus pyogenes or Bacillus anthrax. These three are examples of gram-positive bacteria. Some gram-negatives could be E. coli or Salmonella or Pseudomonas aeruginosa. That's a gram-negative. And you have probably heard of these names before. You've heard, oh, somebody has caught Salmonella, or the person has been diagnosed with E. coli or Staphylococcus or whatever. Now you will always remember that, oh, these actual bacterias that we have names for are actually differentiated to being either gram-negative or gram-positive. And if they are put into these different categories, I can't approach them clinically the same way. So it's not, oh, this is a bacteria and this is a bacteria, give them antibiotics and it's resolved. It's more sophisticated. It's more which type of antibiotic and why. What are we targeting? And to make sense of this, you have to understand first that these are categorized into different groups. And then these groups have different structures. And due to these structures, they will adapt and behave and respond differently to whatever drug we give them, etc., etc. Now, let's indeed mention gram staining. So gram staining is a procedure where we apply crystal violet, which is the primary stain. We then add iodine to fix this stain. Use alcohol to wash it and decolorize it all. We then add safranin, which is the counter stain. When we have done all of that, we look at the results. Again, gram-positive turns purple or violetish. And that's because it retains crystal violet. And this is the high-yield question. Why is it retaining it? Or how is it retaining it? Exactly, because of the thick peptidoglycan layer. And then we turn around and say, what about gram-negative? Why is it turning pink or reddish? because it gets stained by the counterstain safranin. So how is the safranin beating the crystal violet? Because, exactly, the peptidoglycan layer in the gram-negative is much thinner. So it cannot retain this crystal violet, and instead safranin takes over. Now, we already touched upon the outer membrane and the lipopolysaccharides. And if you remember now, we are talking about gram-negative bacteria, right? It has this outer membrane, first of all, that the gram-positive does not have. And expressed on its outer layer, it had these lipopolysaccharides. And you can see here an illustration again. This is the outer membrane, right? And this is the LPS expressed on it. Now, the structure is lipid A, which, if you remember, was an endotoxin, causes inflammation and fever. It has a core polysaccharide, an O antigen, so it has antigenic variability. And the function of this membrane and LPS is to protect against bile salts, against antibiotics, against our immune system. Remember, it can be resistant to phagocytosis. Now, the clinical relevance here is that LPS is a virulence factor in E. coli and, for example, Salmonella. So all in all, gram-negative bacteria have an extra outer layer acting like a shield, so it protects it, making it harder to destroy and kill. But also, it contains LPS, lipopolysaccharides, which is a molecule that causes severe inflammation or can cause severe inflammation and fever in humans when these bacteria infect us. Now, capsules and slime layers. So capsules are like invisible cloaks, right, for these bacteria. It helps them to hide from our immune system. And it makes the infections harder to treat. Now, what are some characteristics of this capsule? Well, it is a thick, organized polysaccharide layer. Its role is to prevent phagocytosis, and it helps in adhesion. An example of this is Streptococci pneumoniae capsule, which enhances virulence. What is the slime layer? Well, it is a thin, unorganized layer aiding in biofilm formation. So, essentially, slime layers are sticky coatings, right? Sticky coatings that help bacteria to stick to surfaces and form a protective communities or something. So biofilms. And the clinical relevance here is that biofilms are resistant to antibiotics. Appendages. So bacteria have tiny structures called flagella or tails, and these are used to swim. Or they have something called pili that are spikes, and this helps them to stick to surfaces. And some pili are even used to transfer DNA between bacteria, helping them sharing tricks like antibiotic resistance, for example. So the flagella structure is of a filament, has a hook and a basal body. Function, indeed, motility and chemotaxis. An example is Heliobacter pylori, which uses flagella to colonize the stomach. We have again then pili. Its role is adhesion to surfaces, conjugation, so transfer of plasmids. Example could be Neisseria gonorrhea, which uses pili to adhere to host tissues. Now, what about the cytoplasm of the bacteria? Well, it's composed of the cytosol, ribosomes, nucleids, inclusion bodies. Now, this is super important. The ribosomes in prokaryotic cells are 70S versus eukaryotic, it's 80S. Now, what does this mean? So first of all, we're talking about ribosomes, and these are essential cellular structures responsible for what? Protein synthesis through the process of translation. You will learn about this in our course on cell biology, but it's worth repeating. So they're important for protein synthesis through translation. And this is where the genetic code from mRNA, messenger RNA, is used to build proteins. Now, these ribosomes, connecting this to this now, these ribosomes consist of two subunits. And we have a large and a small subunit, which together form the ribosome's overall structure. And the ribosome's size, okay, its size is measured in something called Svedberg units. And the unit used is indeed S. So this is 70 Svedberg unit, and this is 80. That's number one. And the Svedberg units indicate the rate of sedimentation during a process called ultracentrifugation. And roughly, they also correlate to the size and the shape. So 70S and 80S refer to the sedimentation rates of the ribosomes in their respective organisms. So the prokaryotic ribosome, 70S, if we start with that, it is composed of two subunits. It has a small subunit called 30S, and it has a large subunit called 50S. And these are composed of different amounts of RNA and proteins, etc. We don't need to go to too much detail here, but the structure is essentially that this 30S subunit, okay, of this prokaryotic is what binds with the messenger RNA to read the genetic code, right? And the 50S subunit is where transport RNA molecules bind and amino acids are linked together to form the growing protein chain. So one example would be E. coli. So this is very directly important and relevant to the whole composition and structure of the bacteria. These ribosomes responsible for protein synthesis in prokaryotic cells, so bacteria, we have 70 Svedberg units. It is then divided into 30S and 50S, and these mean something. They're also very relevant later for treatment processes, but here we know then that 30S was what binds to the messenger RNA to read genetic code, and 50S is where the molecule bind and amino acids are linked together to form the growing protein chain. Now, what about the eukaryotic cells? Here we have 80 Svedberg units, right? Now, 80S ribosomes in eukaryotes are larger than prokaryotes, and they consist of their small subunit is 40S, and its large subunit is actually 60S. And similar here to the prokaryotes, the 40S subunit is what binds to the messenger RNA to read the code, and the 60 subunit forms the site for protein elongation. And the eukaryotes is what we find in humans. Prokaryotic is in bacteria. Now, why does this matter? Why did we jump into the structure of the bacteria, focusing on ribosomes, differentiating between their Swedberg units, finding their subunits, differentiating between 30 and 50S versus 40 and 60? Why does this matter to us as medical doctors? Well, listen, the difference in ribosome size between prokaryotes and eukaryotes is medically significant because antibiotics have evolved to target bacterial 70S ribosomes without affecting the larger 80S ribosomes in human cells. So there has been discoveries whereby we are able to attack this group without attacking this group. So this is key for their effectiveness for antibiotics in treating infections without harming the patient's own cells. So let's just talk briefly about some examples. We have an extensive lecture on antibiotics, but now just to give you an overview. So we have a class of antibiotics called aminoglycosides, and these contain, for example, gentamicin, streptomycin, maybe you've heard of them. Now these aminoglycosides, they bind to the 30S subunit, preventing the attachment of the amino acyl tRNA, thereby inhibiting protein elongation. Excuse me, that's not right. What they do is they bind to the 30S subunit of the bacterial ribosome, causing misreading. That's what they do. They cause misreading of the mRNA inhibiting protein synthesis. If you do like I did, which is for a moment they're confused what these two did, you will then think that the antibiotic we use targets the wrong one. But we had to correct ourselves, right? And say that no, we remember that 30S subunit is what is involved in reading the messenger RNA for inhibiting protein synthesis. And the 50S was involved in the process of the leading to the elongation of the protein chain. So the aminoglycoside group, like streptomycin, will bind to 30S, causing a misreading of this messenger RNA and therefore leading to an inhibition of the protein synthesis. So it can't produce more, right? Another group of antibiotics are tetracyclines. For example, doxycycline. Now what they do is they also bind to the 30S subunit. But what they do is they prevent the attachment of this amino acyl tRNA, thereby indeed inhibiting protein elongation in the bacteria. Another group we could mention is macrolides or macrolides, like erythromycin. What do? They bind to 50S subunit of the bacterial ribosomes, preventing formation of peptide bonds during protein synthesis. No, actually scratch that. We just committed the same error again. The macrolides bind to 50S, yeah, that's correct, but they prevent them from, they are preventing the elongation of the protein chain. The other group is chloramphenicol. It inhibits 50S subunit, of bacterial ribosomes, preventing the formation of peptide bonds, indeed, during protein synthesis. So to summarize then, we have 70S ribosomes. Actually, let's clear this up and say we have 70 Svedberg unit ribosomes found in prokaryotic cells, so bacteria. And these are, number one, smaller than eukaryotic. And they are composed of 30S and 50S. Now, don't be surprised when you encounter this kind of question in your medical exams, because this is indeed questions that will resurface many times through your medical studies. So it's worth mentioning here and repeat. Furthermore, why is it different from eukaryotic? Because eukaryotic had its small subunit of 40S and 60S. This can sometimes be a bit confusing because you see 80 and you think that this is the sum of these two, but it's not. And 70S, I guess 70 is not a sum of 30 and 50. This is just one of those things where you just have to go with it. And another high yield thing was that this smaller subunit was involved in the messenger RNA process, essentially binding to the messenger RNA to read genetic code. And the large subunit is where the tRNA molecules bind and amino acids are linked together to form the growing protein chain. And it's similar in prokaryotic as in eukaryotic. We also learned that antibiotics are developed to target these different subunits, leading to prevention of different processes. And the whole purpose of all of this and knowing all of these details medically is that this difference between them, between the prokaryotic and the eukaryotic, is leveraged to develop antibiotics to selectively target bacterial ribosomes and minimizing damage to human cells, ensuring the effectiveness of treatments. So that's that. Another thing worth mentioning then is the nucleode, which is also here present. You can look at this illustration here in the cytoplasm, and it contains the bacterial chromosome, which is single and has circular DNA structure. Why is it clinically relevant? Well, plasmids carry antibiotic-resistant genes. So that's something we need to keep in mind as we continue developing antibiotics as ways to get around it. Now, what are endospores? These are dormant, highly resistant structures formed under stress. Some examples can be anthrax or botulism, Clostridium botulinum. And some bacteria can hibernate, indeed by forming these tough shells called endospores. And like we said, dormant, highly resistant. So these spores can survive extreme heat or dryness or chemicals, etc., making them hard to kill. And what they do is, very sneaky, they are waiting until conditions improve to then get out and become active again. So this resistance is part of that thing. Now, why is this clinically relevant? Because indeed, they are difficult to eradicate. So we require high-level disinfection to deal with it. Now then, we mentioned in the beginning that we will talk briefly about the mechanisms of transport, because bacteria also needs to bring in food and get rid of waste, and they use these different gates in their membrane to move things in and out. And some bacteria even use these gates to actually pump out antibiotics, making them resistant to treatment. Now, we're just going to briefly mention a couple of mechanisms. So we have different types. We have passive transport and active transport mainly. In passive transport, what is important to remember is that energy is not required for the utilization of these transport systems, while in active transport, energy is used. So, in passive transport, molecules move, right? Molecules move across the bacterial membrane without using energy, without using ATP. And it's driven mainly by concentration gradients, for example, from high concentration to low concentration. And it's important for bacteria because it allows it to efficiently take up small molecules like oxygen and CO2 without expending energy. And this is critical for survival. Then we have active transport. Now, in active transport, the bacteria move substances against their concentration gradient. So from here out, let's say. And this requires energy, usually in the form of ATP. And this is essential for taking up nutrients like sugars, amino acids, and ions in environments where these are present at lower concentrations than inside the cell. It also allows bacteria to expel harmful substances like antibiotics or waste products. This contributes to antibiotic-resistant mechanisms. We have another one we didn't mention here, but it's called group translocation. And it is a unique transport mechanism where the substance is chemically modified during its transport across the membrane. And it allows the bacterium to trap essential nutrients like glucose inside the cell in a modified form that can't easily leave, ensuring the bacterium can use these for their metabolism. So efficient transport is medically incredibly relevant because it influences how bacteria obtain nutrients, expel toxins, survive in hostile environments, including in the presence of antibiotics. Now, what about bacterial cell division? So the process of bacterial cell division is binary fusion. And we have these steps here. And you should try and remember these. So first, we have DNA replication, right? And the chromosomal segregation is happening during the same time. Now, after that, we have septum formation. And finally, we have cell separation. And the time required for a bacterial population to double is called generation time. For example, E. coli divides every 20 minutes under optimal conditions. So to summarize, bacteria reproduce by splitting into two, like copying and pasting themselves. The process is called binary fusion, and it's incredibly fast. Like some bacteria, for example, E. coli, can double every 20 minutes. That's why infections can grow very quickly if we don't treat them. Now, clinical relevance then. We've already touched upon a lot of these things before, but let's repeat. So understanding bacterial structure informs us about antibiotic targets. Peptoglycan, are we targeting the layers for synthesis? Are we targeting the ribosomes, subunit, which one, etc.? We need to be aware of the mechanisms of resistance, like the bacteria using efflux pumps, the lipopolysaccharide, essentially whatever it is repelling our immune system from destroying the bacteria, or when we give it antibiotics, what throws it out of the bacteria? These pumps, for example. We have mentioned the importance of diagnostic techniques to be able to identify, is it a gram positive? Is it a gram negative? And if you remember, we used crystal violet. And if crystal violets remained due to the thick layer, it indicated then a gram positive. And some clinically relevant drugs would be beta-lactams, like penicillin. They inhibit peptidoglycan synthesis. We had another group called aminoglycosides. These were targeting bacterial ribosomes. So in summary then, bacterial cell structures are diverse and specialized for survival. Each structure contributes to pathogenicity, survival, or antibiotic resistance. Understanding these structures help us design diagnostic tools and better therapeutics. And that's the end of this lecture. Hope it was interesting. Continue now to Chapter 3. Welcome to another lecture by MedicoMedics, Learning Made Easy, Microbiology and Infectious Diseases, Chapter 3, Microbial Genetics. So in this somewhat lengthy lecture, we will present microbial genetics, discuss bacterial chromosomes, plasmids, DNA structure, DNA replication in bacteria, concepts like transcription, translation, genetic variation, horizontal gene transfer, transposons, and end with a summary. Microbial genetics is the study of how microorganisms inherit and express genetic information. Why is it important? Because it lays the basis of pathogenicity and antibiotic resistance. It is key to genetic engineering and biotechnology. Types of genetic material in microorganisms include chromosomal DNA. This is core genetic material. And extrachromosomal DNA, like plasmids and transposons. Now let's begin with the bacterial chromosome. It is a single, circular, double-stranded DNA molecule, differing from the linear chromosomes found in eukaryotic cells. And it is located in the nucleoid. So that's a region within the bacterial cell that is not enclosed by a membrane. Ergo, it is not membrane bound. And unlike eukaryotic cells, bacteria do not have a nucleus. And that's high yield. So the bacterial chromosome is found in the nucleoid, a region within the bacterial cell that does not have, it's not enclosed by a membrane. And unlike the eukaryotic cell, bacteria do not have a nucleus. Now, since the chromosome is actually quite large relative to the bacterial cell itself, it is very tightly packed. And supercoiling is the primary method of this compaction, ensuring that the chromosome fits within this small cell. And this supercoiling is maintained by topoisomerases. Let's go with that. And these are specialized enzymes which regulate supercoiling by winding and unwinding the DNA as needed for replication and gene expression. Now, what are plasmids? These are small, circular, extrachromosomal DNA molecules, and these exist independently of the bacterial chromosome. And these plasmids often carry genes that provide bacteria with selective advantage. For example, or including antibiotic resistance, like R plasmids. These encode resistance genes against multiple antibiotics. They also have virulence factors that enhance bacterial pathogenicity. For example, toxins produced in E. coli. And then we have different types. So we have F plasmids, that stands for fertility plasmids, and these enable bacterial conjugation, allowing direct transfer of genetic material between bacteria via something called a sex pilus. We also have R plasmids. These are resistance plasmids, and these carry genes that provide resistance to antibiotics, making infections more difficult to treat. Examples include, for example, plasmid-mediated beta-lactamase production. This is where bacteria acquire enzymes that degrade beta-lactam antibiotics. And beta-lactam antibiotics are, for example, penicillin or cephalosporins. So this then leads to drug resistance. Now, why is this relevant to you as a medical student or someone interested in the subject? Well, understanding plasmids is crucial in clinical microbiology and infectious disease management because they play a significant role in antibiotic resistance and bacterial pathogenesis, and of course, therefore, in our treatment strategies. Now let's go deeper and discuss DNA structure. So DNA is a double helix structure composed of two complementary strands. And we have the base pairings of which have very specific rules, where A is paired with T and G with C. And A stands for adenine, and it's paired with T, that stands for thymine. You have G for guanine and C for cytosine. Now, these strands are anti-parallel, meaning they run in opposite directions. So one strand is oriented 5 to 3, while the complementary strand runs from 3 to 5. Now, let's see the differences between bacterial DNA and eukaryotic DNA. So let's first look at structure. Now, bacterial DNA is typically circular, while eukaryotic DNA is linear and organized into chromosomes. Furthermore, bacterial genes generally lack introns. These are non-coding sequences, introns, whereas eukaryotic genes contain introns and exons. We have more detailed discussions of these in our cell biology courses, but for now, just pick up on that there are some key differences. So there are no introns in bacterial genes, while for humans, we will find introns and exons. Furthermore, eukaryotic is linear, right? The DNA is typically linear, while in bacteria, it is typically circular. And what about packaging? Well, bacterial DNA, as we just mentioned in our previous slide, is supercoiled within the nucleoid, while eukaryotic DNA is wrapped around histones and enclosed in a nucleus. That was one high-yield difference. Bacteria does not have a nucleus. Also, its chromosome is not membrane-bound. All of these are high-yield things for you to pick up on. Now then, let's take a look at DNA replication in bacteria. So we have semi-conservative replication, whereby each daughter DNA molecule or each daughter strand consists of one parental strand. So it receives one parental and one newly synthesized strand. This ensures genetic continuity. Now, steps involved here are initiation. This begins at a specific origin of replication and is a unique sequence where replication machinery assembles. Then we have the enzyme helicase, which unwinds the double helix, creating a replication fork. Then we have something called single-strand binding proteins that stabilize this unwound DNA. After initiation, we have elongation, whereby DNA polymerase synthesizes new strands. I think, if my memory serves me right, it's DNA polymerase 3 that is involved in synthesizing new DNA strands in the 5 to 3 direction. And it uses the parental strand as a template. Now we also have the enzyme primase, which synthesizes RNA primers to initiate replication. So after that, we move to termination, and this is where replication ends at the terminus. So replication continues until it reaches this site, the terminus site. And here we have something called TUS proteins that help halt the replication machinery. Now, why is all this clinically relevant? Well, for example, in the development of antibiotics, so some antibacterial drugs inhibit bacterial DNA replication. For example, quinolones. Quinolones like ciprofloxacin target DNA gyrase, which is a topoisomerase, preventing supercoiling and leading to bacterial death. Because here, during termination, the two daughter DNA molecules are separated by topoisomerase 4. Yeah, topoisomerase 4. And this ensures for proper segregation. However, knowing the clinical relevance here is that these medicines like ciprofloxacin targets this DNA gyrase, the topoisomerase, and inhibits it. And this leads to the prevention of supercoiling and therefore bacterial death. So understanding bacterial replication is critical in antibiotic resistance mechanisms and in the development of new antimicrobial therapies. We could also mention rifamycins like rifampin, another antibiotic which inhibits RNA polymerase. So it blocks bacterial transcription. So all in all, knowing these different steps and what happens during these different steps, we can develop antibiotics to stop it. Similar to using antivirals in virus, for example, where we can, by understanding the cycle, the viral replication cycle, we can intervene to block at different stages. And it's the same here. Now, we mentioned in the previous slide rifampin, and we said that it blocks or inhibits RNA polymerase. So why is it clinically relevant? Let's connect the dots. So transcription is the process of converting DNA into RNA. So it's the first step in gene expression where we are converting genetic information in our DNA into functional molecules. And the steps you may recognize from the previous slide, we have initiation. This is where RNA polymerase binds to the promoter region of a gene. This leads to the DNA unwinding, exposing the template strand. Then we have elongation, where RNA polymerase reads the DNA template in the 3 to 5 direction and synthesizes a complementary mRNA strand in the 5 to 3 direction. So RNA polymerase reads the DNA in 3 to 5. And then it has synthesized a complementary mRNA, right? So RNA polymerase synthesizes mRNA complementary in the 5 to 3 direction. And the RNA sequence follows the base pairing rules that we mentioned before, where adenine pairs with uracil instead of thymine, because we're talking about the RNA sequence now. When we talked about DNA, we said that adenine was paired with thymine. All right? But now it's adenine pairing with uracil. And cytosine pairs with guanine. Now, we reach the termination phase. So transcription stops at the termination signal, or at a termination signal. And the newly formed mRNA detaches from the DNA. Now, some key features we can mention to differentiate between bacterial and eukaryotic transcription are the following. So in bacteria, this is occurring in the cytoplasm. Okay? And mRNA is immediately translated. But in eukaryotic cells, transcription is occurring in the nucleus. And mRNA undergoes processing before translation. These processes include, for example, splicing and capping. So again, we repeat the difference between the bacteria and eukaryotic cells, between prokaryotes and eukaryotes. As we mentioned early in the lecture, it wasn't membrane bound, if you remember. It didn't have a nucleus. The bacteria cell doesn't have a nucleus compared to the eukaryotic cell. And so here we also see differences during the process of transcription. Here it happens in the cytoplasm. While in eukaryotic cells, transcription occurs in the nucleus, and bacteria doesn't have a nucleus. Hope it all made sense. Now let's talk about the next step, translation, following from transcription. So translation is the process of converting messenger RNA into a functional protein. And this occurs in the ribosome, where amino acids are assembled in a specific order dictated by the mRNA sequence. And the ribosome 70S, or S, if you remember from previous lectures in this series, stood for Swedberg unit. So 70S in bacteria, remember we're talking about bacterial cells now. If it's eukaryotic cells, we would say 80S. Okay? So in bacteria, ribosome 70S facilitate translation by reading mRNA codons. And tRNA, which stands for transfer RNA, carries specific amino acids, carries or delivers specific amino acids that correspond to these codons. in the mRNA. And the genetic code is universal, with each codon specifying a particular amino acid. Now let's look at the steps. So again, we have initiation, elongation, and termination. So during initiation, the ribosome assembles in the start codon, which codes for methionine in bacteria. The small ribosomal subunit binds to mRNA, followed by the large ribosomal subunit. And the subunits are in bacteria, if you remember, the small one is 40S, the large one is 60S. Did you fall for that? Because that's not correct. It's actually 30S, small, and 50S, large one. And this is the ribosome 70S. It's in eukaryotic cells where we have 40 and 60, and we call it 80S. Now, don't get caught up in the mathematics here. The sum of this is not this, but let's leave that for now. That's one of those medical mysteries that we just let slide by us. Now, let's go back. So, we talked about initiation. Now, during elongation, what happens is that the ribosomes move along the mRNA, reading codons sequentially. Now, transfer RNA molecules deliver these amino acids, which are joined by peptide bonds. So these amino acids are bound by peptide bonds, or joined actually by peptide bonds, forming a growing polypeptide chain. And the ribosome has 3 sites for transfer RNA binding. It has something called A site, which stands for amino acyl site. And we have the P site, which is the peptidyl site. And then we have the E site, the exit site. And essentially in the A site, the new transfer RNA carrying an amino acid, right? Here you can see in the illustration, the transfer RNA carries this amino acid and brings it in. And in the P site, so we can call this A here, and then in the P site, the peptide bonds form between amino acids. And in the exit site, the empty transfer RNA, remember, it has delivered something, now it's empty. It leaves then the ribosome. Then we arrive at the termination. This is when a stop codon is reached. Translation then stops. A release factor binds, causing the polypeptide chain to detach from the ribosome. So when this is completed, now it detaches. And the ribosome then disassembles, and the newly synthesized protein folds into its functional shape. Now, as always, let's put it in some clinical context. So examples would be tetracyclines inhibit bacterial translation by targeting ribosomes. But how? Well, it targets ribosome 70S. And it prevents protein synthesis from occurring. So this makes then translation a key target for antibiotic therapy against bacterial infections. And we have specific antibiotics targeting 30S, 50S, etc. Now, even though this is not a lecture on antibiotics specifically, for those who are interested, let's go a bit further and specify a bit more so that you understand why you are learning details like subunits for ribosomes. Why are we differentiating between 30S and 50S, for example? Now, let me put this in some clinical context. So, we talk about antibiotics. And I mentioned briefly just now that there are antibiotics targeting 30S and there are those targeting 50S. So, in the big picture, these antibiotics have something in common, which is inhibiting translation at early stages. And it's essentially interfering with mRNA decoding or with the ribosomal function in general. So we have a group like tetracyclines, and the antibiotics typically found here are doxycycline, for example. And this prevents transfer RNA binding to the A site of the ribosome, blocking protein elongation. Do you remember when we said A site and then we had the P site and the E site? I used to memorize this as APE. Don't ask me why, but it worked. Now, once you understand that, oh, there is an A site, right, where it's bringing the protein for delivery, and then we have the binding site, etc., and then the empty transfer RNA leaving. So by understanding at this level, we get that tetracyclines are actually preventing the tRNA binding to this A site, blocking elongation. So we can't get peptide bonds forming between these amino acids. Ergo, we will not be producing these proteins. So we say that these tetracyclines are bacteriostatic. They inhibit bacterial growth without killing them. We also have another group targeting the 30S subunit, which is aminoglycosides. Some of these are gentamicin or streptomycin, etc. These bind to the 30S subunit, causing misreading of mRNA, leading to the production of non-functional proteins. And these are bactericidal, so these kill the bacteria. And now, just to make it complete, we also said we had 50S subunit. So an antibiotic targeting the 50S ribosomal subunit is, for example, macrolides. These antibiotics generally inhibit peptide bond formation, also preventing protein synthesis. So now we're somewhere else, right? The forming of the peptide bonds. We mentioned that during the P. So it inhibits it here. The other one was blocking the binding site at A. So now it's hopefully beginning to become interesting and relevant to you. Why these details matter? Why are we studying these different subphases? Precisely because we have drugs targeting them differently, leading to different results, etc. And these macrolides, maybe you've heard of erythromycin or azithromycin or clarithromycin. All of these bind to the 50S subunit, preventing translocation of the ribosome along the mRNA. And these macrolides are typically, they are bacteriostatic. So to recap, we had antibiotics targeting the 30S ribosomal subunit. And these typically inhibit translation at early stages. We had aminoglycosides and we had tetracyclines. Then we said we have antibiotics targeting the 50S subunit. And these antibiotics generally inhibit peptide bond formation or ribosomal translocation, also preventing protein synthesis. And here we mentioned macrolides. Excellent. Let's keep going. Now, it's also important to mention genetic variation in bacteria. So we can have mutations, and we have different types of mutations. For example, point mutations, like silent, missense, or nonsense. Here, a single nucleotide change in the DNA sequence that leads to different effects. So if it's a silent mutation, there is no effect on the protein. If there is a missense mutation, it alters one amino acid in the protein, potentially changing its function. If it's a nonsense mutation, it introduces a premature stop codon, leading to a truncated, non-functional protein. And then we have insertions and deletions. So insertion essentially means addition. You're adding something, right? And deletion is removal. So removing something. So it's either an addition, an insertion, or removal, a deletion of nucleotides. And causes of these changes could be spontaneous or induced by mutagens like UV light. So, for example, resistance to rifampin could be caused or is caused by mutations in RNA polymerase. Now, what is horizontal gene transfer? It is the exchange of genetic material between bacteria, occurring independently of cell division. So it is the transfer of genetic material between organisms not involving reproduction. Now, this process plays a crucial role in genetic diversity, adaptation, and the spread of antibiotic resistance among bacterial populations. Now, what is the mechanisms? So first we have transformation. It is important to know that it is the uptake of naked DNA from the environment. So bacteria absorb free DNA fragments from their environment and integrate them into their genome. And this requires the recipient bacterium to be competent, meaning capable of DNA uptake. In the Griffith experiment, for example, with Streptococcus pneumonia, the experiment demonstrated transformation where a non-virulent strain acquired capsule genes from a heat-killed virulent strain, making it then pathogenic. So something that was not virulent acquired something, a capsule gene, from something dead, and then became itself pathogenic. So transforming. Then we have transduction. So a bacteriophage, for example, which is a virus. Some people get confused because it contains the word bacteria, but it's bacteriophage. So a bacteriophage, a virus, infects a bacteria, incorporating bacterial DNA into its viral genome and transferring it into another bacterium. So we have two types of transduction worthy of mentioning. One is generalized transduction. So a random bacterial genes are transferred. And then you have specialized transduction, where only specific bacterial genes near prophage insertion sites are transferred. So example, in Corynebacterium diphthera or diphtheria, it acquires diphtheria toxin genes via bacteriophage infection, and it makes it virulent. Then we have conjugation, which is direct cell-to-cell transfer via pilus. So via the pilus, it's a direct transfer between two cells. And so it requires direct contact between the bacterial cell via this sex pilus. And it involves the transfer of plasmids, if you remember, extra chromosomal DNA, plasmids. Example, Our plasmid transfer in E. coli enables the spread of antibiotic resistance genes, making it harder to to treat this infection. So why is all of this treatment related or clinically relevant? Well, horizontal gene transfer, HGT, is a major driver of multi-drug resistance in bacteria. For example, methicillin-resistant Staphylococcus aureus, MRSA or MRSA, acquires its resistance genes via HGT. Another is virulence factor transmission. Why is it, again, clinically relevant? Because bacteria can gain toxins and other pathogenic traits, increasing their ability to cause disease. Now, genetic elements, transposons. So transposons, or they are also called jumping genes, are these mobile genetic elements that can move from one location to another within the genome. And unlike plasmids, for example, they cannot replicate independently, but they can integrate into chromosomal DNA or plasmids. And an example of a function is that many transposons carry antibiotic resistance genes, allowing bacteria to survive antibiotic treatment. Another thing they're involved in is that some transposons contain genes encoding toxins or virulence factors, which enhances the pathogenic potential of bacteria. So an example of transposon is in vancomycin-resistant Enterococcus, also abbreviated VRE. Here, transposons have played a critical role in spreading vancomycin resistance among these species, making it more difficult to treat this infection. So in summary then, bacteria have efficient genetic systems enabling rapid adaptation. Relevant concepts from the lecture included 70S ribosome, and if you remember, for eukaryotic cells we had 80S, and for prokaryotic, like bacteria, we have 70S. And we have the subunits, 30 and 50. And other important concepts, the absence of a nucleus. So eukaryotic cells have a nucleus. Bacteria do not. We mentioned transcription and translation, and it's high yield to remember the initiation, elongation, and termination processes. Antibiotic treatment targets many times specific subunits, like tetracyclines target subunit 30S and macrolides 50S. Horizontal gene transfer drives evolution and resistance. And that's the end of the lecture. Thank you for listening. Continue now to Chapter 4. Welcome to another lecture by MedicoMedics, Learning Made Easy. Microbiology and Infectious Diseases, Chapter 4, Virology, the study of viruses. So in this lecture, we will present what are viruses, talk about its structure, classification, the viral replication cycles, pathogenesis of viral infections, host defenses against viruses. What are some medically important viruses? Further on, we will talk about some antiviral drugs, some vaccines, and end with a summary. Now, viruses are these tiny infectious agents made of genetic material, DNA or RNA, and they are encased in a protein coat. Some key features of viruses include that they cannot replicate without a host cell, and they are much smaller than bacteria. Examples of viral diseases are the flu, HIV, COVID-19, etc. Now, what is the structure of a virus? So the basic components include a capsid. This is the protein shell that protects the genetic material. We have nucleic acid, so DNA or RNA containing viral genes. And then we may have an envelope. This is a lipid layer with spikes for host attachment. Now, viruses come in different shapes. So we have helical, like the rabies virus, icosahedral, like the poliovirus, and complex shapes like bacteriophages. So viruses are like tiny packages. The genetic material is the instruction manual, right? The genetic material contained is the instruction manual, and the capsid is the box protecting it. Now, some viruses may add this extra layer, the envelope, which has these spikes to help it essentially attach to the host, to go into the host cell. And here you have an illustration. So these are the spikes, which it may or may not have. It has either DNA or RNA inside. We have the capsid protecting it, and then the envelope itself. Now, how do we classify viruses? We do it based on genetic material. Is it a RNA virus or a DNA? Is it enveloped or non-enveloped? What is its host specificity? Animals, plants, or bacteria? Now, examples can be RNA virus like influenza, DNA virus like herpes virus. Then we have retrovirus like HIV. So classifying viruses is like sorting packages by their type and contents. Some carry DNA, others RNA. And some even come with tools to actually rewrite their host's DNA, like HIV. Now let's talk about the viral replication cycles. And we will use as an example HIV. So the steps here are important and high yield to remember. What happens first is attachment. So the virus binds to the host cell. Then we have entry. So the virus injects its genetic material or enters entirely. After that, we have replication. So viral genes take over the host cell machinery to make new viral components. Then we have assembly, so new viruses are assembled inside the cell. And finally, release, so the host cell bursts, also called lysis, or releases viruses gradually. And what is important here is that antiviral drugs often target specific steps. That's why you need to remember all of these steps. So here in this illustration, we have the HIV virus here. And first, it's binding, right? And then it goes through entry, fuses in. The virus capsid, through something called reverse transcription initiation, continues on to get to the nucleus of the host cell. And here, uncoding occurs, whereby the viral DNA has now successfully been delivered into the host cell. After that, it integrates with the host cell genetic material and then starts producing viral mRNA, continuing on to produce whatever it's producing and then exiting through assembly and release. Now, we will, in our lectures on HIV specifically, dive into greater details, talking about each of these steps in relationship to the HIV virus itself. However, here, what is high yield are remembering these steps. Remembering that the virus will first bind, then it will enter or deliver its material. Then that material will go through replication and essentially taking over the host cell. Following from that, it will start producing whatever it is encoding or coded to produce or programmed to produce. And then it will start leaving the cell to go spread, causing damage. And another way to think about it is that viruses are like burglars. So they break into the host cell, use the resources to copy themselves, and then they leave. And often when they leave, they damage or destroy the host cell in the process. Now, what is the pathogenesis of viral infections? So, how viruses may cause disease, either killing or damaging host cells. This is called cytopathic effects. Another way is triggering excessive immune responses, so inflammation. And even though inflammation is fundamentally a good thing, if it's too prolonged, too excessive, it may actually start causing damage. And we have latency among viruses. So we have dormant viruses that reactivate later, like, for example, herpes. So you get it, and then it's quiet for a long time, and then it activates. Some examples of how these viruses may cause disease. So HIV destroys our immune cells. And if you don't have your immune cells, you will be immunodeficient. And when that happens, other pathogens that exist that would have normally been repelled by your immune system now attack your body and eventually overwhelming your system, even leading to death. And another example is influenza that damages our respiratory cells. Now, naturally, throughout evolution, our body has developed different ways to combat and defend against bacteria, against viruses, etc. So we have our innate immunity helping us against viruses. The first line of defense, for example, interferons and natural killer cells. When these fail, we have our adaptive immunity, whereby we have our antibodies that bind and neutralize viruses. And then we have T cells that help destroy infected cells. We use vaccines, of course, to stimulate immunity to prevent infection. And we go into depth in our classes on immunology, talking greatly in detail about all of these different cells. But for now, it's important to keep in mind that, indeed, vaccines are used to stimulate our immunity. We have an adaptive immune system helping us to fight off viruses. We even have our innate immunity trying to do that. When it fails, we have treatment, antivirals to do that. So let's mention just a few medically important viruses. We have the DNA viruses like herpes virus, causing cold sores and shingles, hepatitis type B virus, leading to liver infection. We have RNA viruses like the influenza virus and COVID-19 or SARS-2 virus. We also have retroviruses like HIV, causing, for example, AIDS. Now, just to put it into some clinical context to make it more memorable, let's dive a little bit deeper into HIV and how it causes AIDS. So what happens is that HIV, all right, it enters the body and it targets our CD4 plus. These are actually called T helper cells. But these are part of our T cells. Remember, we mentioned we have our T cells and we have B cells. And one of these T cells is the CD4, also called the T helper cell. Now, HIV specifically targets the CD4 cell. And let me just mention that this cell is incredibly vital and important in our adaptive immune system, or in our immune system in general. So what happens is that HIV binds to it.
and it binds to receptors on this CD4 and co-receptors on the cell surface. So the CD4 expresses, for example, let's see if my memory serves me right here, but we have CCR5, CXR, CXCR4. Now, these are co-receptors on this T helper cell. So the HIV binds to these. And when it does that, it then fuses with the cell membrane and releases, right? It releases its RNA into the host cell. So we talked about the first part was attachment and then entry, right? So it fuses and releases its RNA into the host cell. Now, what happens then? So the integrated DNA, viral DNA, now uses the host cell's machinery to produce new viral RNA and proteins. And these components then assemble into new HIV particles. And what happens then is that as HIV then continues to replicate, it will damage and destroy infected CD4 T cells. And furthermore, our immune system also attacks infected cells. So further destruction of CD4 T cell happens. So not only is the virus itself causing this damage, but our already existent immune system is identifying infected cells and it starts attacking it as well. So your immune system is now under great siege. Now, over time, what happens is that the progressive loss of CD4 will weaken the immune system overall, because I mentioned it's a very important immune cell involved in the totality of your adaptive immune system in general, important specifically for signaling, telling your immune system where to go and what to do, etc., etc. We are going into detail in our course in immunology, but for now, take my word for it, this is an important cell. So what happens is that as CD4 counts go down violently, the body then becomes unable to fight off opportunistic infections. And what then happens is that we have the development of AIDS. So AIDS is diagnosed actually, right? AIDS is diagnosed when CD4 T cells drop below a certain number. And I will not mention this number because it's not relevant right now, but when our CD4 cells drop below a certain level, or when specific opportunistic infections or conditions escalate, right? And so what happens is that without treatment, this will lead to severe immune suppression and indeed life-threatening complications. So very opportunistic pathogens or causes of infection that would have normally been repelled if you had a normal CD4 count. You would have thrown these away, let's say. All of these opportunistic, all of these pathogens now become opportunistic. So they will seize the moment. So let's mention briefly some antiviral drugs. So what is the mechanism of action in general here? Well, they can block virus entry. Remember the steps again. We first had attachment and entry. So a mechanism of action of some antiviral drugs would be to block this entry. Like, for example, fusion inhibitors for HIV. We can use drugs to inhibit replication. So remember, when it's entered the host cell and delivered its content, now it wants to replicate. So we give a drug to stop this. Furthermore, after replication and all of that, what does the virus do? It begins to move towards releasing its product to go cause more mayhem. So we have drugs to block that, preventing virus release. Now, the challenge is that viruses mutate quickly, which leads to drug resistance. So research continues to find and try to stay a step ahead, but it's difficult because these are quite clever. But this is all in all what you need to remember. So you need to repeat and revise the steps of the viral replication cycle and then understand, first of all, what these different cycles mean and what is happening during these different stages, but also then understand how our medical interventions actually work here. We stop here, or we stop it here, or we stop it here, etc., etc. Now let's talk about viral vaccines. So we have weakened viruses used to stimulate our immune system, to train it essentially, like the MMR vaccine, the mumps, measles, and rubella vaccine. We can use killed virus, like in polio vaccine. We have mRNA vaccines, like the COVID-19 vaccine. And what are some benefits? Well, preventing infection and reducing disease severity. And another challenge here would be that mutating viruses may require updated vaccines, for example, the flu. So vaccines are like training exercises for our immune system. We are presenting it with a little part of this pathogen, enough to stimulate our immune system to start building protection and antibodies, essentially, cells, so that in the future, whenever this thing appears again, we already have a protection against it made in our system. But not enough is given to stimulate disease. Now, this isn't always bulletproof, but that's the general idea here. Viruses and biotech. So, for example, in gene therapy, viruses are used as vectors to deliver therapeutic genes. In vaccine development, we're using viral platforms. In cancer therapy, oncolytic viruses are used to kill cancer cells. So scientists can use viruses for good by turning them into delivery vehicles for medicines, vaccines, or even cancer treatments. So essentially, it's like using a hijacked enemy tool to help people. So that's one way to stay ahead of the game. But viruses can be very clever and even recognize what we are trying to do sometimes, so they find new ways to adapt and create further problems. And this is why all of this is very interesting. So in summary then, viruses are unique. They are not alive, but they are capable of causing diseases. This is very philosophical. And understanding their structures and life cycle indeed helps us fight infections. Advances in virology are improving treatments, vaccines, and biotech. So viruses are strange and fascinating. Indeed, they are not alive, but they can wreak havoc or even be used for good. So by studying them, we can protect ourselves and also unlock new medical advancements. And that's the end of this lecture. Thank you for listening. Continue now to Chapter 5. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 5, Mycology, the study of fungi. So in this lecture, we will do an introduction to fungi, discuss structure, classification, some infections, diagnosing fungal infections. We will mention antifungal agents, and then continue down to end with a summary. So fungi are eukaryotic organisms with cell walls made of chitin, And chitin is a natural polysaccharide made up of acetylglucosamine, essentially a derivative of glucose molecules linked together. And it's primarily known for its role as a structural component in various organisms. And in fungi, in the cell walls of the fungi, it provides with structural integrity. Now let's mention some types of fungi. So we have yeasts, right? These are single-celled fungi. An example is Candida. We have molds, so multicellular fungi with filamentous structures like Aspergillus or Aspergillus. Then we have dimorphic fungi. So this switches between yeast and mold forms like histoplasma. Now, why is this important? Because these are decomposers in ecosystems, and they are producers of medicines like penicillin. So all in all, fungi are like recyclers of nature. They're breaking down dead things, and they come in different forms like single cells, yeast, or filaments, molds. And some of them are used to make medicine, others may cause infections. Now let's look at the structure of fungi. So key features, we mentioned a cell wall made of chitin. It's not made of cellulose like in plants. And we have hyphae, so filamentous structures that form mycelium, the fungal body. Another important feature are spores. So these are reproductive units for spreading and growth. And we have types of hyphae. So we have septate, divided by walls, and choanocytic, where we have no internal walls. Now, how do we classify fungi? Well, we have these major groups: Zygomycota, Mycota, bread molds like Rhizopus, Ascomycota, sac fungi like Candida or Aspergillus, Basidiomycota, club fungi like mushrooms, and let's say that's Cryptomycota, aquatic fungi with flagellated spores. And the classification is based on reproduction. So either we have sexual reproduction, so fusion of spores, or asexual reproduction, so spores produced by mitosis. So what's important to remember is that fungi are grouped based on how they reproduce. Now, fungal infections or mycoses. These can be subdivided into different categories. So we have superficial mycoses. These affect hair, skin, and nails, like athlete's foot. We have subcutaneous mycoses affecting deeper skin layers or tissues. Systemic mycoses, so affecting internal organs like histoplasmosis. And opportunistic mycoses affecting immunocompromised people like Candida albicans. Transmission, direct contact, or inhalation of spores, or environmental exposure. So fungal infections range from annoying skin problems to serious internal infections, especially in people with weak immune systems. And these infections often spread, right, through contact or breathing in of these spores. Now, how do we diagnose fungal infections? Well, the methods we use include microscopy, so we do staining and viewing under a microscope. Culture test, growing fungi on a special media. We do molecular techniques like PCR to detect fungal DNA. Some examples like the co-test, dissolving human cells to highlight the fungal cells, the India ink stain detecting Cryptococcus in cerebrospinal fluid. Now, let's talk about some antifungal agents. And antifungal drugs target the unique parts of the fungi, like their walls or membranes. But treating fungal infections can be tricky because their cells are a lot like ours. So treatment needs to be very careful and specific. But what kind of types do we have? Well, we have azoles or azoles. These inhibit cell membrane synthesis, like fluconazole. We have polonies. These bind to fungal membranes, causing cell death. We have something called echinocandins. These inhibit fungal cell wall synthesis. And if you remember, if we talk about challenges, right? And it says fungal infections are harder to treat than bacterial ones due to similar cell structures to human. Do you remember some specific things in bacteria that makes it in a way easier to target and to differentiate from human cells? Well, if you remember in our lecture, we talked about the ribosomes that are involved in the creation or production of proteins, right? And that we identified in bacteria, we have different subunits compared to eukaryotic cells. So these prokaryotic cells had the smaller subunit of 30S. If you remember, S was Swedberg unit, and 50S, compared to humans that had 40S and 60S. That's not an S. And that eukaryotic cells are put into 80S in total, and bacterial is 70S. And this differentiation helps us in developing drugs to specifically target these different things without actually targeting human ribosomes or preventing human synthesis of proteins, etc. Now, in fungi, this is a bit tricky because we don't have this same differentiation, so it is indeed harder to treat. Fungi and the environment. Ecological roles. So fungi are nature's sort of cleanup crew. They break down dead things and help plants to grow. But they're also sort of troublemakers in agriculture, specifically causing plant diseases that can actually ruin crops. But some roles include decomposers, indeed recycles nutrients by breaking down dead matter. Symbiosis, so it partners with plants for better nutrient absorption. And of course, pathogens, so it causes diseases in plants. And now let's turn to fungi in medicine. So applications. We use it in antibiotics. An antibiotic known to most people is penicillin, and it is derived from penicillium, a fungi. We have immunosuppressants like cyclosporine, used for organ transplants, used in vaccines like Candida albicans, used for research in antifungal vaccines. Then we have fungal toxins, aflatoxins. So these are harmful substances produced by Aspergillus. So all in all, fungi are both sort of heroes, but also villains when we're talking about medicine. They give us life-saving drugs, like antibiotics, but also produce harmful toxins that can contaminate, for example, our food, etc. A bit of a yin and yang of nature. Some industrial applications of fungi. So it's used in fermentation, yeasts in bread, beer, and wine production. Bioremediation or remediation, breaking down pollutants in the environment. enzymes, so industrial production of cellulases and proteases. What about fungi and biotech? Some applications include producing biofuels from plant waste, involvement in genetic engineering. Let's say that as a model organism, the production of recombinant proteins like insulin. So in summary then, fungi are diverse and important in ecosystems, in medicine, and in industry. While some fungi are beneficial, like penicillin, others can cause serious infections or even crop damage. Understanding fungi helps us fight diseases and use them in innovative ways. So fungi are everywhere, essential for life, but can also cause problems. By studying them, we can harness their power for good while protecting ourselves from harm. And that's the end of chapter 5. Continue now to chapter 6. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, chapter 6, Parasitology, the study of parasites. So in this lecture, we will give you a general introduction to what are parasites, different types, its life cycles, the pathogenesis of parasitic infections, and we will mention transmission, diagnosis, treatment, preventing infections, some examples of parasitic diseases, and then move on towards a summary. Now, parasites are organisms that live in or on a host organism, obtaining nutrients at the host's expense. And we have different types of parasites. So we have protozoa. These are single-celled parasites. For example, Plasmodium. And why is Plasmodium interesting? Because it causes malaria. We have helminths. These are worm-like parasites. Examples, Ascaris, tapeworms. We have ectoparasites. These are external parasites like lice or ticks. Now, importance. Causes significant diseases in humans, animals, and plants. So, parasites, all in all, are these uninvited guests that live off their host, often making the host sick. Some, as we mentioned, live inside the body, like worms or protozoa, while others stay outside of the body, like lice or ticks. And why are these interesting? Because these are, for example, ticks spread Lyme disease. So all of this is quite relevant. Now, these rely on their hosts to survive, but usually they harm them in the process. Now, let's continue talking about these different types. So we said protozoa. These are microscopic, single-celled organisms, illustrated here. Examples, as we mentioned, Plasmodium leads to malaria. We have Giardia, which causes diarrhea. Then we have another type, helminths. These are multicellular worms, right? And the types could be roundworms or flatworms, like tapeworms, etc. Then we have these ectoparasites that live on the skin or outer surfaces. Examples, ticks, which spread Lyme disease, and fleas, causing itching and transmit infections. And what's interesting is that, indeed, parasites come in different forms, right? Tiny single cells, like Plasmodium, to larger worms, like roundworms, or even these creepy crawlers, like ticks. But each type of these has a unique way of surviving, so it causes problems for its host. Now, let's talk about the life cycles of parasites. So we have, first of all, stages. where we have the egg becoming a larva and then developing into an adult. This is a prime example of the pathway for helminths. Then we have trophozoites. So the egg becomes a trophozoite, and then continues to become a cyst. This is a pathway for protozoa. Another important concept is hosts. These are differentiated into definitive host and intermediate host. So in the context of parasites, the term host refers to the organism in which the parasite lives and from which it derives its nutrients or protection. And these hosts are categorized based on their role in the parasite's life cycle. So when we talk about a definitive host, this host is the organism in which the parasite undergoes sexual reproduction. And this host is critical for the completion of the parasite's life cycle. So, for example, in schistosoma, humans serve as a definitive host because the adult worms live and reproduce sexually in the human bloodstream. All right? Now, what about intermediate host? The intermediate host is the organism in which the parasite undergoes asexual reproduction, right? So no sexual reproduction, but rather asexual reproduction or other developmental stages, but it does not do it sexually. So it is essential for the parasite's development and transmission to the definitive host. So, for example, again, for schistosoma, snails are the intermediate hosts, where the parasite undergoes larval development, and so understanding life cycles is indeed crucial for controlling infections, specifically due to the way these sustain and develop. Now let's turn our attention to the pathogenesis of parasitic infections. Some mechanisms of harm include the stealing of nutrients. So, for example, tapeworms absorbing food from our intestines, damaging tissues like Plasmodium destroying our red blood cells. Another is triggering immune responses, so severe inflammation from schistosoma eggs. And what could be some symptoms following it? Well, malnutrition, or anemia, fever, organ damage, etc. Now, what about the transmission of parasites? So some modes of transmission include ingestion, so through contaminated food or water like Giardia or tapeworm eggs. We have vector-borne spread, so spread by insects like mosquitoes, leading to malaria. Skin penetration, so larvae entering through skin, like schistosoma. Direct contact, so close contact with infected individuals or surfaces, for example, lice. Now, let's make this somewhat boring lecture a bit more fun and clinically relevant. So let's try and connect different parts of the lecture thus far. So we talked about, for example, mosquitoes, right? And let's start with the mosquito and let's end with malaria, or the process of having malaria. So what happens is that this mosquito that is itself infected with something called Anopheles mosquito, or it's called that, bites a human being, okay? And it injects spirozoites into our bloodstream. Then these spirozoites or sporozoites enter our liver, and they infect our liver cells, also known as hepatocytes. Okay? And what happens is that then they then multiply to form something called merozoites. So from sporozoites to merozoites. And these merozoites are then released into our bloodstream. Now these merozoites infect our red blood cells. All right? And they multiply and cause these red blood cells, okay, to rupture. And this releases then more merozoites and toxins into our blood because they were multiplying, remember? Now, the rupturing, this is where it becomes clinically important because the rupturing of these RBCs occurs in several cycles and lead to these symptoms of fever, for example, chills, sweating. These are classical malaria symptoms. And this continuous RBC destruction leads to eventually severe anemia. So essentially, we have now a reduced oxygen carrying capacity of our blood. Why? Because oxygen transports through blood and binds to hemoglobin in our red blood cells. So, what are then the complications of all of this? Well, the accumulation of parasitized RBCs eventually blocks capillaries, causing even organ damage, leading to, for example, cerebral malaria. And eventually, severe anemia or organ failure or cerebral complications, all of these could lead to death. So what started as something very innocent, a tiny little mosquito, a mosquito bite, spread and multiplication, entering into our blood cells, multiplying and rupturing, leading to fever, leading to red blood cell loss, leading to anemia, leading to severe anemia, eventually even leading to death. So that's a big picture of the why parasites and the study of parasites, etc., is crucial. Now, this was just an overview. We go into the details of the malaria in our malaria lecture. But for now, hope you get a brief overview of why we're studying this, why it's important to know the mode of transmission in a parasite, to know it's vector-borne, that it's spread by insects like mosquitoes for malaria. Beautiful. Let's go on. Now, let's briefly mention some ways to diagnose parasitic infections. So methods include microscopy, so identifying eggs, larvae, or protozoa in stool, blood, or tissue samples. Serology tests, so detecting antibodies against parasites in our blood. Molecular methods like using PCR to detect parasite DNA. Some practical examples could be a blood smear, so identifying Plasmodium in malaria, or a stool test finding Giardia cysts or helminth eggs. And then what is the treatment of parasitic infections? So we use drugs like antiprotozoal drugs. These treat protozoan infections like chloroquine for malaria. Anti-helminthic drugs like albendazole, these are used to treat worm infections. What are some challenges we're confronted with? Well, drug resistance in parasites. Difficulty in treating parasites that hide inside our cells. So, all in all, treating parasites involves medicine that either kill the parasite or stop it from reproduction or reproducing. But indeed, some parasites do develop resistance or hide in places where medicines can't easily reach. Then some ways to prevent parasitic infections include, for example, hygiene, so hand washing and proper sanitation, safe food and water, cooking food thoroughly, avoiding contaminated water, vector control, so using insect repellents or bed nets for mosquito-borne diseases, vaccination. However, it is limited in availability, but malaria vaccines are, for example, in development. Now, let's just repeat some examples of parasitic diseases and mention some new ones. So, under protozoan diseases, we have indeed malaria caused by Plasmodium, transmitted by mosquitoes. We have amoebiasis caused by Entamoeba histolytica, leading to diarrhea. Then we have helminth diseases. So we have ascariasis. These are large intestinal worms. Schistosomiasis caused by blood flukes. We have ectoparasitic diseases like Lyme disease spread by ticks. Now let's just briefly touch upon the connection between fungi and parasitology. So some parasitic organisms have fungal-like characteristics, like Pneumocystis jirovecii in immunocompromised patients. So treatment and diagnosis often overlap with fungal infections. So in summary then, parasites are diverse and can cause significant harm to humans. Understanding their life cycles, mode of transmission, and treatment is crucial for prevention. Global efforts are making strides in reducing parasitic infections. And that's the end of Chapter 6. Continue now to Chapter 7. Welcome to another lecture by MedicoMedics, Learning Made Easy, Microbiology and Infectious Diseases, Chapter 7. Immunology Basics, How the Body Defends Itself. So in this lecture, we will present the immune system, talk about innate immunity, adaptive, antibodies and their roles, vaccines. We will talk about inflammation and the immune response, immunology in everyday life, recap, and then end with a summary. So introduction to the immune system. Now, the immune system is the body's defense system against harmful microorganisms or cancer, toxins, etc. Its key roles include recognizing harmful invaders, attacking and neutralizing these invaders, and furthermore, to remember past infections for faster future responses. So the immune system is a highly trained security team that guards our body. It looks for threats, it neutralizes them, but it also keeps a record of it so it can respond even faster if the same threat shows up again. Now, briefly, some components of the immune system. So, organs and tissues that are important. We have the bone marrow. This is where blood cells are produced, including our immune cells. We have the thymus. This is where T cells maturate. We then have lymph nodes and our spleen. These are involved in filtering harmful substances and activating immune responses. Cells here include white blood cells. These are the soldiers of the immune system. And the types of these cells include lymphocytes, so our T cells and B cells. Also our macrophages and our neutrophils. Now let's begin with innate immunity. And before we move on, this is a course in microbiology, and of course, immunology is a part of it. However, we have a dedicated course on immunology that we recommend you taking a look at some point to go into details of all of this. But let's carry on. So our innate immunity, we have physical barriers. That includes, first of all, our skin. This is a first line of defense against the entry of pathogens. So things that want to come in and cause damage, our skin repels or protects us against. We also have mucous membrane that trap microbes in the respiratory tract and in our digestive tracts. So these are some physical barriers. Then we have chemical barriers like our stomach acid. We have enzymes in our saliva and our tears. These also help break down and repel pathogens. But then we have cellular defenses. These include phagocytes like macrophages that engulf and destroy invaders. We also have natural killer cells that target infected or cancerous cells. So the innate immunity, as suggested by the name innate, you're born with it, is like the body's first responders. So the skin and mucus are barriers, while specialized cells, like in our cellular defenses, act like firefighters. So they go to the site of an infection to eliminate invaders quickly. Now, the second type is adaptive immunity. And the name is very suggestive because it's adaptive. So it adapts to something. It learns, let's say. And this is our second line of defense. Key features here includes specificity. It targets specific pathogens. In comparison to our innate immunity, where our immune cells are up and about, they notice a pathogen, and then they may get activated to engulf and destroy it. But they are not specifically targeting anything, compared to our adaptive immune system, where these cells target specific pathogens. Another key feature is memory. These cells remember pathogens for a faster response in the future, and we will talk about how that happens. And the main players in our adaptive immune system are B cells that produce antibodies to neutralize invaders, and we have T cells. These kill infected cells and coordinate immune responses. So our adaptive immunity is like a personalized defense system. It learns from experience. It remembers past threats, and then it develops very precise weapons to fight off specific invaders. And these precise weapons that are developed are called antibodies. So how the immune system works together. Let's look at a step-by-step process. First, we have recognition. Immune cells identify the invader using different receptors. That then leads to activation, so signals trigger a response. Inflammation, which is where the recruitment of immune cells occur. Then we have the attack, so pathogens are neutralized by, for example, phagocytosis from our macrophages or antibodies from our B cells, or through direct killing. Then we have resolution. So we've gone from recognition, activation, attack, resolution. Immune responses wind down, preventing damage to the body. Because as you may understand, when there are these reactions that are in a way positive, right? We are fighting off something that's not supposed to be in our body. But this process itself, the process of inflammation, can become very dangerous if it's out of control. Now, antibodies and their roles. So we mentioned that our B cells produce these proteins that bind to specific antigens. Specific antigens means markers on the pathogens. So this pathogen here has something, right? It has a specific antigen on it. And these B cells that have been produced with these antibodies are built to bind specifically to this and destroy this cell. Now, functions then. Neutralizing toxins and viruses. So essentially this. Mark pathogens for destruction by other immune cells. So this cell, let's say we have another cell here that finds this one, signals it, puts something on it, and says, okay, now a signal will be transmitted. So another cell here, it catches this signal and moves to destroy it. What about vaccines then? So how do vaccines work? Well, we introduce harmless parts of pathogens. Now that we just mentioned how the immune system on a very fundamental level works. We have memory cells where we produce antibodies. These then recognize future threats when the same thing is presenting to attack it. With this knowledge that we have these memory cells, what do we do then in relationship to vaccines? Well, we manipulate the system by introducing harmless parts of this pathogen, of these antigens that are expressed on the pathogens, and we introduce it to our body so that our immune system recognizes it and starts producing antibodies to it. But it's important to understand harmless is key here. So we introduce enough of this pathogen to trigger our immune response to start producing these antibodies for future use, but not enough to produce actual pathology. So the purpose, to generate memory cells for faster future responses. And when we talk about vaccines, we have live vaccines where we use a weakened virus, for example, in the MMR vaccine, which stands for measles, mumps, and rubella. We have inactivated vaccines where we use killed virus, like in polio. We have mRNA vaccines like COVID-19 vaccines. So vaccines are like practice drills for your immune system. They show your body what a pathogen looks like, so it's ready to fight it when the real one shows up, so to speak. Now let's briefly talk about disorders of the immune system. Here is an example of an overactive immune response. Like in allergies, this is when our immune system is reacting to harmless substances like pollen. So something that's actually not a pathogen that needs to be destroyed. It is harmless if it enters our body. Nevertheless, for some reason, our immune system is recognizing it as a threat. So we have an immune response and the increase of inflammation and whatever symptoms then follow. Now imagine you have a huge influx of this harmless substance. You will have a strong immune response, meaning a strong allergic reaction that is not necessarily justified, other than that your immune system has wrongly decided to target this thing. But nonetheless, from a medical standpoint, we still have to treat it, because this immune response, this inflammation, could actually lead to fatal consequences. Like, for example, whatever this allergen is that is not dangerous leads to fluid buildup or constriction or vasodilation in your whatever, let's say in your respiratory tract, like asthma, right? And you get these constrictions and you can't get enough air in. We have something called anaphylaxis, anaphylactic shock. You cannot breathe, so you could die. So we have treatments for these allergies, like using antihistamines, etc., etc. So this is just an example of our immune system overworking itself and not necessarily related to an actual opportunistic pathogen. Similarly, we actually have a subject where we study autoimmune diseases. And this is where the body is attacking its own cells, like rheumatoid arthritis. So similarly here, that this pollen is not dangerous, but our immune system is attacking it, and it's the response that is actually destroying or causing harm. The same way, we have, for whatever reason, our cells have been taught to attack this cell right here. And this cell might be our fluid in our joints. And it's actually there for protective reasons. we want this synovial joint fluid to be there because it facilitates less friction between your joints, right? You have a bone here and you have a bone here. And then you have this fluid here that helps these two from hitting each other. But for some reason, your immune system has developed autoimmunity, and whatever these fluids are built out of, or whatever they consist of, those cells have become the target of our immune system. So it will over time destroy this fluid. And then patients will present with, for example, rheumatoid arthritis. Now, autoimmune diseases are many, and we cover them in immunology and in other lecture series. But this is another prime example of how our immune system is dysfunctional. We can also have the opposite. So we talked about overactive immune response. We can have a weakened immune response. We call it immunodeficiency, like having HIV. So the idea here is HIV as a virus enters into its host cells and it targets specifically. We're not going into detail and you don't need to memorize this, just an idea. We talked about T cells and we talked about B cells, which make up the main parts of our adaptive immune system. Now, these T cells, you will learn, is mainly divided into what we call CD4 and CD8 T cells. And these T cells, the CD4 type, are the prime target of this HIV virus. So it connects to it and destroys them. And these CD4 cells are vital in the coordination of our immune system. So over time, what happens in HIV is that these CD4 cells go down. The count goes down. And over time, when we say the patient has developed AIDS, is that otherwise non-dangerous, let's say, otherwise non-dangerous pathogens that our immune system normally is able to control and repel because of the loss of these immune cells. Over time, this ability to repel goes down. And as it goes down, the amount of opportunistic pathogens, so pathogens that could actually then cause damage, increase. And this becomes overwhelming and could eventually even lead to death. Now, inflammation and the immune response. So the body's response to injury or infection is inflammation, and it is marked by redness, swelling, heat, and pain. And the idea or the purpose of inflammation is to contain and eliminate the threat and to promote healing. But you can have chronic inflammation, where you have a long-term inflammation that keeps going on, like an arthritis. And what may be a good thing to start with, inflammation is a positive thing, with the purpose of eliminating the threat, containing it, and promote healing. But if it continues over time, this causes tension, stress, and over time actually leads to damaging consequences rather than healing ones. So, for example, think of it like this. Inflammation is like a fire alarm, right? You have a fire alarm in your house. You want that thing to start beeping when there is a fire. So it's alerting the body that there is a problem and it sends help to put this fire out. But pretend the alarm just keeps going. What would happen if it just constantly keeps going? What would happen to your daily life? Would it not be disrupted? Would you not go crazy? Would you not feel like you need to jump? So the idea is similar here. Prolonged something of a good thing could actually turn very bad. It's both philosophically interesting, but it's also physiologically quite interesting. And it develops a whole different side to medicine, where we need to prepare not only for developing something to resolve a problem, but potentially developing something to resolve the consequences or potential consequences of that something that is solving a problem. And the thing goes on and on. Moving on then to the immune system in cancer. So immune cells, indeed, detect and destroy abnormal cells. Tumors develop ways to hide from our immune system. This is why cancer is one reason why cancer is so tricky, because we do have immune cells developing. We have the presence of different immune cells ready to destroy these abnormal cells that are a consequence of uncontrolled cell division, so cancer. But the problem is tumors develop different ways to hide from this. So it maintains its division, and our immune system is just looking at it without actually having any reaction to it. So what we try to do is we try to use immunotherapy, or we use essentially the immune system to fight off the cancer. So we find ways to trigger our immune system to accelerate its response and to attack these abnormal cells. So it tries to find ways to hide and evade us. We try to find ways to essentially identify it still. So staying one step ahead is part of what research is doing. So in summary then, the immune system protects against infections, cancer, and toxins. It has two main layers, or parts. We have the innate immune system, our general defense, that is non-specific. And we have the adaptive immune system, which is specific. We use vaccines, antibodies, and therapies, and all of these rely on understanding how the immune system works. And that's the end of this lecture. Thank you for listening. Continue now to Chapter 8. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 8, Host-Microbe Interactions. In this lecture, we will present host-microbe interactions. We will talk about the normal microbiota, opportunistic pathogens, briefly talk about virulence factors and host defense mechanisms, some beneficial microbe-host interactions, dysbiosis of this microbia, clinical implications, and end with a summary. So, introduction to host-microbe interactions. What's the definition here? Well, it's interactions between a host, so human or animal, and microorganisms, so bacteria, a virus, fungi, or parasites. Now, what are the types of interactions? So if it is mutualism, it means both the host and the microbe benefit. For example, gut bacteria. If it's commensalism, microbe benefits without harming the host. And if it's parasitic or parasitism, the microbe benefits at the host's expense. So what's the normal microbiota? Microorganisms that live on and inside the body without causing harm is what we typically call the normal microbiota. What are the roles here? It aids in digestion and in producing vitamins, for example, E. coli in our gut. It prevents colonization by harmful microbes. It helps stimulate the immune system. Some examples can be in our skin, we have Staphylococcus species. In our gut, we can find Lactobacillus, Bifidobacterium, etc. So these normal ones are like a built-in defense system and some kind of a helper team. They all have positive benefits. Now, let's mention opportunistic pathogens. These are normally harmless microbes that may then cause disease under certain conditions. Examples could be Candida albicans. It causes infections when the immune system is weak. or Clostridium difficile causes severe diarrhea after antibiotic use. So the triggers here could be immune suppression or disruption of normal microbiota, for example, caused by the use of antibiotics. Now, the pathogenesis of infectious diseases. What are the steps? First, we have entry. So microbes enter the body through skin or respiratory tract or other routes. It then attaches itself and adheres to host cells using specialized molecules. We then have invasion, where microbes spread within the body. After that, we have evasion, so the microbes escape our immune system. So you can assume that in between invasion and evasion, our immune cells try to respond to this, but for whatever reason, they escape the immune system. Once they've done that, then we come to damage. So the microbe cause tissue destruction, or there is an immune reaction, but it's an overreaction, leading to further damage. And understanding these different steps and targeting these steps will help prevent or treat infections. What is virulence factors, or what are virulence factors? Well, these are traits that make the microbe more capable of causing disease. For example, if they have adhesion molecules, these will help the microbe to stick to the host cells. It's toxins, so poisonous substances that damage host tissues. Enzymes, these might help microbes invade tissues. So targeting virulence factors can weaken these pathogens. So expressed in a different way, virulence factors are like tools or weapons that these microbes use to invade and harm our body. And like we mentioned, some of these weapons are, for example, adhesion molecules. So it helps it to stick to our cell. And others is releasing toxins, damaging, causing great damage to our cells. Or ways to evade our immune system, for example. So these are virulence factors of a certain microbe, let's say. What are the host defense mechanisms? We mentioned this in the previous lecture, but just for the sake of brief repetition, let's go through them again. So we have physical barriers like our skin or mucous membranes. We have enzymes that help in breaking down and repelling pathogens, our skin, for example. We have innate immunity, which is our rapid response. So we have phagocytes like macrophages or neutrophils, and these then produce inflammation, leading to the containment of the invasion and also promoting healing. Then we have our adaptive immunity, which is a long-term protection where we find our antibodies produced by our B cells and our memory cells, which helps us with faster responses in the future. And the way microbes might evade these immune mechanisms could be by using capsules, antigenic variation, or hiding inside the host cell, etc. Now, what about some beneficial microbe-host interactions? Examples could be gut microbiota. And here we produce short-chain fatty acids for gut health. Also prevents in harmful bacterial growth. In our skin, it protects against pathogens by out-competing them. Another is that friendly microbes help train the immune system. So it's the old saying, keep your friends close, but your enemies closer. Microbial dysbiosis. So this is the imbalance in the normal microbiota. We said that these are your friends or enemies that you keep closer, but if it becomes imbalanced, So we have a dysbiosis. It could lead to negative effects. So, for example, causes of developing a dysbiosis could be antibiotic overuse. It could be poor diet. You could have it due to chronic diseases like diabetes. And what happens is some consequences could be increased risk of infections, like Clostridium difficile could suddenly become more prone to start attacking. And we also link a dysbiosis to obesity, mental health issues, autoimmune diseases, etc. Now, some emerging research in host microbe interactions include the Human Microbiome Project, where we are mapping all microbes in the human body. We're looking at links between gut microbes and brain health, so the gut-brain axis, its role in chronic diseases like diabetes and heart disease. Therapies are being developed like probiotics, the use of probiotics, introducing beneficial microbes to our body. Use of fecal microbiota transplants to help restore gut balance. Now, briefly, some clinical implications then. Diagnosis. So, identifying infections based on microbial interactions like stool tests for gut infections. Antibiotics for harmful microbes. We use probiotics to restore balance. And of course, a key way to prevent all of this is vaccination and hygiene. So antibiotics would fall under treatment, more specifically. Probiotics would fall under preventive measures. So in summary then, microbes and humans have a complex relationship. Some help, others harm. Maintaining balance in the microbiota is key to health. And emerging therapies focus on leveraging beneficial microbes. And that's the end of this lecture. Continue now to Chapter 9. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 9, Principles of Sterilization and Disinfection. In this lecture, we will present an introduction to sterilization and disinfection, to mention some methods, some processes, disinfection methods, discuss antiseptics versus disinfectants, and then continuing down, we will mention some common misuses of disinfection and end with a summary. Now, sterilization means the complete elimination of all microbes, including spores, while disinfection is the reduction of harmful microbes to a safe level. Importance here is to prevent infections in healthcare settings, to ensure safety in food and water. So, sterilization and disinfection are like deep cleaning for microbes. Where sterilization removes all microbes, disinfection makes things safe by essentially reducing their number. Now, some methods. We have some physical methods of sterilization, like heat. It is the most common and most effective method. So, moist heat through the process of autoclaving is 121 degrees Celsius under pressure. And it kind of works like a pressure cooker. Then we have dry heat, like hot air ovens. Another way is irradiation. We're using UV light or gamma rays. Filtration, removing microbes from liquid and air. So, sterilization uses tools, essentially like heat or light, or even chemicals. For example, it's not mentioned here, but we have ethylene oxide gas, and it sterilizes delicate instruments that we use. But anyway, sterilization uses tools like these: heat, radiation, light, or chemicals, indeed, to destroy microbes. Autoclaving, the gold standard in sterilization. An autoclave is like a super-powered pressure cooker used in hospitals and labs to make tools completely germ-free. It's quite fast to do, it's effective, and very reliable. Now, how it works is it combines high temperature, so 121 degrees Celsius, and pressure of 15 pound per square inch psi. And it uses this combination of these two, so high pressure and high temperature, to kill all microbes and spores. Some applications of this, well, sterilizing surgical instruments, lab glassware, and other things. Limitations. It's not suitable for heat-sensitive materials. Now then, moving on from sterilization to disinfection methods. So we have types of disinfectants like alcohol, for example, ethanol and isopropanol. These are effective against bacteria and viruses. We use chlorine compounds like bleach. It kills a broad range of microbes. Quaternary ammonium compounds, so quats. These are used for surface cleaning. Hydrogen peroxide, effective on hard surfaces. Some applications of these: cleaning hospital surfaces, instruments, and household items. Now, let's talk antiseptics versus disinfectants. So, antiseptics are used on living tissues like skin. Examples include alcohol, iodine, chlorhexidine. And antiseptics are gentle cleaners, safe for our skin. Now, compared to antiseptics, we have these disinfectants, which are stronger and meant for cleaning surfaces like floors and countertops, etc. So they are used on non-living surfaces. Again, antiseptics, living tissues like skin, on humans. Disinfectants used on non-living surfaces. Example include bleach and quats. And clinical relevance: antiseptics used for wound care and disinfectants for cleaning surfaces. Now let's mention some levels of disinfection. So we have high-level disinfection, where we kill all microbes except some spores. Example, glutaraldehyde for surgical instruments. We have intermediate-level disinfection, where we kill bacteria, viruses, and fungi, but not spores. Example, alcohol. Then we have low-level disinfection. These reduces microbial load, less effective against some bacteria and viruses. Example, quats for general cleaning. Now, what are some factors affecting disinfection and sterilization? Key factors include microbial load. So the more microbes, the harder to sterilize. The type of microbe, so spores and viruses, are more resistant. Contact time, longer exposure improves effectiveness. Another factor is material compatibility. So some materials can't handle heat or chemicals. So all in all, the effectiveness depends on number of germs, the type of germs, how long the cleaner stays in contact with it, and some items need special care indeed to avoid damage. Microbial resistance to sterilization and disinfection. So more resistant microbes include, for example, prions. These are infectious proteins, and it needs extreme heat to get rid of, like 132 degrees Celsius. Bacterial spores require autoclaving. And if you remember, autoclaving was the combination of high temperature and high pressure. The least resistant: enveloped viruses like influenza, vegetative bacteria like E. coli. Now, sterilization and disinfection in healthcare. So applications: sterilizing surgical tools, catheters, and syringes. Disinfecting hospital rooms and high-touch surfaces. Another application is in preventing healthcare-associated infections. So proper sterilization reduces the risk of healthcare-associated infections, like, for example, MRSA or methicillin-resistant Staphylococcus aureus, if my memory serves me right. Now, common misuses of disinfection. Examples can include overuse of disinfectants leading to microbial resistance, improper dilution of cleaning agents, not following contact time recommendations. Consequences, well, reduced effectiveness and an increased risk of infections. This sort of makes sense. So in summary then, sterilization eliminates all microbes. Disinfection reduces them to safe levels. Different methods are used depending on the situation and the material. Proper use is critical to preventing infections and maintaining safety. And that's the end of this lecture. Continue now to Chapter 10.
Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 10, Antimicrobial Agents. In this lecture, we will give you an overview over antimicrobial agents, how these work. We will talk about antibiotics, antiviral agents, antifungal, and antiparasitic. Furthermore, we will mention antimicrobial resistance, how to deal with this, and future developments in antimicrobials, and end with a summary. Now, introduction to antimicrobial agents. So these are substances that kill or inhibit the growth of microorganisms. And we have different types. So we have antibiotics. These target bacteria. Antivirals. These target viruses. Antifungals, targeting fungi, and antiparasitics, targeting parasites. Now, as we mentioned, antimicrobial agents are drugs designed to target and eliminate pathogens while minimizing harm to the host. They are classified, as we mentioned, based on the type of microorganism that they act against. So let's start with antibiotics. These target bacteria. So antibiotics are drugs that inhibit bacterial growth, and we call these bacteriostatic, or kill bacteria. We call these bactericidal, by targeting essential bacterial processes. So some mechanisms of action is, for example, inhibiting cell wall synthesis. And an example of a penicillin is, the example is penicillins, also known as beta-lactams. And these include also, the group of beta-lactams also includes cephalosporins and carbapenems. So these inhibit cell wall synthesis. Then we have antibiotics that inhibit protein synthesis. For example, tetracyclines, macrolides, aminoglycosides. These all inhibit protein synthesis, which means they target somehow the ribosomes that are involved in synthesizing our proteins. If you remember from other lectures, we talked about subunits 50, subunits 30, etc. Then we have antibiotics that disrupt nucleic acid replication, like fluoroquinolones or rifampin. We also have antibiotics that alter membrane permeability, like polymoxins. Now, some clinically relevant information could be that overuse of antibiotics leads to antibiotic resistance. For example, MRSA, MRSA, or multidrug resistant tuberculosis, for example. And some antibiotics are broad spectrum, so they target many different bacteria, while others are narrow spectrum, so they target specific bacteria. Then we have antivirals. Now, since viruses rely on host cells to replicate,
antivirals interfere with viral replication without harming the host cell. So some mechanisms of action include inhibiting viral entry or viral attachment, like miraviroc for HIV, oseltamivir for influenza, for example. Then we have antivirals that block viral genome replication, like acyclovir for herpes or remdesivir for COVID-19. We have antivirals that prevent viral protein processing, like protease inhibitors for HIV or for hepatitis C. We have antivirals that stimulate our immune system, like interferons for hepatitis B and C. Now, many viruses mutate rapidly, leading to drug resistance, like HIV, influenza, etc. And unlike antibiotics, most antivirals are virus-specific and do not have broad-spectrum effects. If you remember, antibiotics have broad spectrum, and we also have narrow spectrum. Moving on to the third type, antifungals. Target organism is, of course, fungi. Now, fungi are eukaryotic organisms, so it makes them harder to target without harming the human cells, because we also have eukaryotic cells. Now, antifungals exploit differences like ergosterol or ergosterol in fungal membranes. So, what would be the mechanism of action here? For example, disrupting the cell membrane integrity with drugs like fluconazole. We also have antifungals that inhibit cell wall synthesis. We have antifungals that block nucleic acid synthesis. Now, the clinical relevance here. So fungal infections can be very superficial, like athlete's foot, or systemic, for example, invasive candidiasis. Candidiasis. I have no idea how to say that. Never learned it. Candidiasis. Let's go with that. And what is important is that immunocompromised patients, like those having HIV or cancer or transplant, etc., are actually at a higher risk for severe fungal infections. So keep that in mind. Finally, then, we have antiparasitics targeting parasites. Now, these are used against protozoa, like malaria, causative agents of Giardia, and also helminths, so worms like tapeworms, roundworms, etc. Parasites, essentially. Now, the mechanism of action here is inhibiting or disrupting metabolism and replication. For example, metronidazole for Giardia, chloroquine for malaria. Another mechanism of action of these antiparasitics is targeting neuromuscular function, like ivermectin, I think is the name, for helminths. And finally, nutrient uptake. So one mechanism of action of antiparasitics could be to affect nutrient uptake, like albendazole for intestinal worms. Yeah, albendazole, this one. Now, many parasitic infections are endemic in tropical regions and require actually long-term treatment. And the development of drug resistance in, for example, malaria, which is caused by Plasmodium falciparum, is a major global health concern. Now let's repeat ourselves. So antibiotics, we talked about two types. Broad spectrum, these target a wide range of bacteria, for example, amoxicillin. And then we have narrow spectrum, these target specific bacteria, for example, vancomycin. And we have different classes of antibiotics. This is just a few of them. We have an extensive course on antibiotics where we go into detail through all of these different classes. But here we mention beta-lactams, so penicillin, cephalosporins, for example. Macrolides, erythromycin, azithromycin. Tetracyclines like doxycycline, aminoglycosides like gentamicin. And these are used for bacterial infections like having pneumonia or urinary tract infections, etc. So antibiotics in general in medicine are used to fight bacterial infections. That's quite obvious now. Now, some target a wide range of bacteria, others are specialized for a certain type. Antivirals then. We had acyclovir, so this treats herpes infections. Oseltamivir, or Tamiflu, treats influenza. Remdesivir treats COVID-19. What is the mechanism? To prevent virus entry into cell, blocking virus replication, inhibiting virus release from infected cells. So the idea is that antiviral drugs are like, say you are on a road here and you're trying to go, these roadblocks ahead make you take turns or be delayed or not even be able to get through. That's kind of what antivirals do. They just block, sort of. They get the virus, it stops the virus from getting into the cell or spreading somewhere else or making copies of themselves. Now, what about antifungal agents? So azoles like fluconazole and itraconazole. We have polyenes like amphotericin B, quiniocandins, caspofungin. You gotta love medicine. So the mechanism here is targeting fungal cell walls or membranes. And finally, anti-parasitic agents. So we have anti-malarials used for malaria like chloroquine. We have anti-helminithics like albendazole or ivermectin. We have antiprotozoals like metronidazole. And indeed, these disrupt parasite metabolism or reproduction. Now, it's all beautiful and lovely that we have treatments for so many things, but microorganisms develop the ability to resist effects of drugs. We call this antimicrobial resistance. And the causes of these are, for example, overuse or misuse of antibiotics. Another one is incomplete treatment courses. Its use in agriculture. And the consequences of this is that it's harder to treat infections, and it increases healthcare costs and indeed mortality. Now, how do we combat antimicrobial resistance? Well, we use antibiotics only when necessary. We make sure that we complete a prescribed treatment when given. We develop new antimicrobial drugs, of course, and we promote infection prevention. Antibiotic stewardship, so coordinated efforts to ensure the appropriate use of antibiotics. Goals are to reduce misuse and overuse, preserve the effectiveness of existing antibiotics, and of course improve patient outcomes. Some challenges in developing new antimicrobials. Reasons for the slow development, its high cost and long timelines. Resistance develops faster than new drugs are created. This is a tricky one. Of course, limited financial incentives for pharmaceutical companies. So what would be some solutions then? Well, incentives for research and investment in alternate therapies. Now, looking forward then, some emerging strategies include phage therapy, so using viruses to kill bacteria. This is a very manipulative, but I like it. CRISPR technique, so gene editing to target resistant microbes. And of course, new vaccines to prevent infections. Focus areas would be to using targeted therapies with fewer side effects. Alternative approaches like boosting the immune system. So in summary then, antimicrobial agents are vital for fighting infections, but misuse leads to resistance. New approaches and careful management are essential for long-term success. And of course, everyone plays a role in combating antimicrobial resistance. That's the end of this lecture. Continue now to Chapter 11. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 11.
Clinical Microbiology Laboratory Diagnosing Infectious Diseases. So in this lecture, we will give you a brief overview of our clinical labs, talk about sample collection and handling, microscopy and microbiology, mention some culture methods, and continue down to discuss some challenges and end with a summary. Now, what do we do in the lab? So the purpose is to identify infectious agents, bacteria, viruses, fungi, parasites. Furthermore, to guide treatment by determining antimicrobial susceptibility, to support infection control and public health efforts. Key activities: processing patient samples like blood, urine, swabs, and conducting tests for pathogens. So microbiology labs act like detectives, figuring out what germs are causing an infection and helping doctors choose the right medicine to fight those. Now, sample collection and handling. So common sample types include blood, urine, stool samples, sputum, and wound swabs. Best practices: proper collection techniques to avoid contamination. Timely transport to lab to preserve sample quality. Challenges: contaminated or improperly stored samples give inaccurate results. Now, some types of microscopy used in microbiology. Well, we have light microscopy, so stains like gram stain for bacteria. Fluorescence microscopy highlights specific microbes under fluorescent dyes. Electron microscopy, high detail for viruses and ultrastructures. So microscopy provides a quick preliminary look at pathogens. Culture methods. So the idea is to grow microbes from patient samples to identify them. Types of media used: enriched media. It supports fastidious bacteria. Example, blood agar. Then we have selective media, where it inhibits some microbes while promoting others. Then we have differential media, helps differentiate microbes based on its appearance, like its color change. Clinical relevance. So culture confirms the presence of pathogens. So culturing is like growing plants in a garden. Different soils, quote unquote, media, help grow specific microbes, and once they grow, we can identify them. Molecular diagnostic techniques. So methods include polymerase chain reaction. This helps us detect microbial DNA or RNA. We have next generation sequencing, provides detailed genetic analysis. The advantages here are that are rapid and highly sensitive and useful for hard to culture microbes like viruses. Serological tests, of course, looking at the blood, helps us detect antibodies or antigens in a patient's blood. ELISA test helps detect antibodies to infections like HIV. Rapid antigen tests help identify infections like strep throat or COVID-19. So it's useful for diagnosing current or past infections. Because indeed, if we find that we have antibodies for something, it means we've probably been infected before by that thing. Antimicrobial susceptibility testing. So the purpose is to determine which antibiotics are effective against a bacterial infection. Methods include Kirby-Bauer test or disc diffusion, broth dilution for minimum inhibitory concentration, MIC. So this guides doctors in choosing the right antibiotic. Now, quality control in microbiology labs. So the importance is to ensure accuracy and reliability of test results. Processes are regular calibration for equipment, running control samples alongside patient tests. Challenges include maintaining high standards with limited resources. So quality control ensures lab tests are accurate, like double checking every step in a recipe to make sure the final dish is perfect. Don't you just love these metaphors? And then some challenges in clinical microbiology include antimicrobial resistance and limited access to advanced diagnostic tools in low resource settings.
We can resolve a lot of these through global collaboration and investment in new technologies. So in summary then, clinical microbiology labs are essential for diagnosing and managing infectious diseases. Advanced tools like polymerase chain reaction and AST improve diagnosis and treatment. Collaboration and innovation are critical to address future challenges. And that's the end of this lecture. Continue now to Chapter 12.
Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 12, Epidemiology and Public Health, Microbiology, Tracking and Preventing Disease. In this lecture, we will cover the topic of epidemiology, discuss some key concepts, chain of infection, types of disease transmission, concepts like outbreak investigation, vaccination and herd immunity, some global challenges, and end with a summary. Now, epidemiology is the study of how diseases spread, who they affect, and how to control them. The objectives include identifying cause and risk factors for diseases, track outbreaks and their patterns, and develop strategies for prevention and control. So epidemiology is like being a detective for public health. Now, some key terms include incidence. This is new cases of a disease in a population over a specific time. Prevalence, total cases of a disease in a population at a given time. Endemic, it's a disease regularly found in a certain area. For example, malaria in parts of Africa. Epidemic, a sudden increase in cases in a specific area. And then pandemic, a global outbreak, like the recent COVID-19. Now, let's talk about the chain of infection. And what are some components here? Well, we have the infectious agent, like a bacteria or a virus. Reservoir, where the microbe lives. So, for example, in humans or in animals. Portal of exit, so how it leaves, like coughing or blood. Mode of transmission, like air, water, or vectors. Portal of entry, so how it enters via the skin or mucous membranes. Susceptible host, for example, unvaccinated individuals. And the idea is to break this chain will prevent diseases. So think of the chain of infection as a sequence. If we can break one of these links, it would be a potential way to stop the disease from spreading. For example, if we're dealing with an airborne transmission, then we can wear a mask. Now, types of disease transmission. So we have direct transmission. This is person to person, like touching or sneezing. Indirect transmission, so contaminated objects like doorknobs, or food or water like cholera. Vectors like mosquitoes spreading malaria. Some tools for epidemiological studies include descriptive studies, so describing patterns, who, where, when. Analytical studies, investigating causes and risk factors, like case control studies. Interventional studies, so test preventive measures, like vaccine trials. The role of public health in microbiology. So functions, identifying pathogens causing outbreaks, tracking antimicrobial resistance trends, develop vaccines and public health strategies. During an outbreak, how do we investigate? So we have the following steps to adhere to. First, confirm the outbreak. Then identify the pathogen and the source. Third, implement control measures like quarantine or vaccination, etc.
and of course communicate findings to the public. Now, vaccination and herd immunity. So vaccination, the idea is to protect individuals by triggering immune responses. The idea here is we use either a killed part of the virus or the pathogen or an inactive part of it, enough to stimulate the person's immune system to start developing antibodies, meaning for the person to get stimulated enough to start producing cells, immune cells. So in the future, if this pathogen presents itself, we already have a large, vast group of immune cells ready to attack it. This is the idea. So herd immunity protects communities by reducing overall spread of disease when enough people get vaccinated. And this is clinically relevant because it's critical for eradicating diseases like, for example, smallpox. So vaccines protect individuals and communities. Herd immunity, specifically, works like a firewall which blocks diseases from spreading when enough people are immune. Some global challenges in infectious disease control, emerging and re-emerging diseases, so zoonotic diseases like Ebola, SARS-CoV-2, drug resistance pathogens like MRSA. Barriers include poor healthcare infrastructure, vaccine hesitancy, climate change expanding vector habitats. So, for example, mosquitoes spreading more throughout the world, carrying malaria is an example of that. So, in summary then, epidemiology helps us understand and control disease patterns. Collaboration and prevention are key to stopping outbreaks. And that's the end of the lecture. Please continue now to Chapter 13. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases. Chapter 13, Emerging and Re-emerging Infectious Diseases. Now in this lecture we will talk about what are emerging and re-emerging infectious diseases, its causes, some examples, some causes of re-emerging diseases, and some examples. Further, we will continue talking briefly about the role of antimicrobial resistance and global challenges ahead, and end with a summary. So let's begin by differentiating between emerging and re-emerging infectious diseases. So emerging diseases are new infections appearing in a population for the first time. Examples: recently we had COVID-19 or the SARS or the Nipah virus. However, when we talk about re-emerging diseases, these are previously controlled diseases that are increasing in incidence. Example: tuberculosis and measles. Now, some causes for emerging diseases. Key drivers include zoonosis, so diseases jumping from animals to humans, like Ebola or COVID-19, as far as we know. Then we have urbanization, so crowded cities make it easier for diseases to spread. Another one is globalization, increased travel and trade spread diseases quickly. And then, of course, climate change, so expanding habitats for vectors like mosquitoes. Now, some examples of emerging diseases. We mentioned COVID-19 caused by SARS-2, spread rapidly globally. The Zika virus linked to birth defects, spread by mosquitoes. Hantavirus spread by rodents, causing severe respiratory illness. Now, what are some causes of re-emerging diseases then? Some key factors mentioned are vaccine hesitancy. It leads to outbreaks of measles and polio. Antimicrobial resistance, so old diseases like tuberculosis become harder to treat. And weak healthcare systems, so unable to maintain control measures. Some examples of re-emerging diseases include TB, tuberculosis, rising due to drug-resistant strains. Measles, increasing due to vaccine hesitancy. Dengue fever, resurging in urban areas with poor mosquito control. The impact of emerging and re-emerging diseases. So health impact, we have high mortality rates in, example, Ebola. Long-term health complications like Zika virus and its relation to birth defects. The economic impact, so we have healthcare costs, loss of productivity and travel restrictions. Some social impacts, we develop stigmas, fear, and misinformation. Surveillance and monitoring, why it matters? Well, early detection prevents outbreaks from spreading. We hope, anyway. Tools, so we have disease surveillance systems like the World Health Organization's Global Outbreak Alert and Responsible Network. Genomic sequencing to track pathogen evolution. So the surveillance is like keeping a watchful eye on diseases. The earlier we spot them, the faster we can sort of contain it. Now, controlling emerging and re-emerging diseases. So strategies include vaccination campaigns, public education to counter misinformation, strengthening healthcare systems for faster response. Examples, polio vaccine eradication efforts, and COVID-19 vaccination programs. Now let's talk about the role of antimicrobial resistance. So the definition here is that germs evolve to resist treatments, making infections harder to treat. In our previous lectures, early in this course, we talked about the bacterial cell wall, plasmids, we talked about viruses and how these can adapt over time in different ways to expel and remove our antibiotic treatments, also ways to evade our immune system, etc. So overall, it's the same philosophy here. Germs evolve to resist treatments, and this makes the infection harder to treat, like multi-drug resistant tuberculosis or drug-resistant malaria. Now, what are some solutions here? Judicious use of antibiotics, and of course, development of new drugs. What about the role of public health in disease prevention? Efforts promoting vaccines and hygiene, enhancing disease reporting and global collaboration, supporting research on emerging diseases. So some global challenges and future directions. Some key challenges include poor access to healthcare and vaccines, climate change driving vector-borne diseases like mosquito spread, for example, leading to the increase of the Zika virus, political and economic instability affecting response efforts, future focus, strengthening global health systems, investing in research and innovation, and addressing social determinants of health. So in summary then, emerging and re-emerging diseases pose significant global challenges. Surveillance, vaccination, and public health efforts are essential to control outbreak. Collaboration and innovation are crucial to address future threats. And that's the end of Chapter 13. Thank you for listening. Continue now to Chapter 14. Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 14, Zoonotic Diseases, Bridging Human and Animal Health. In this lecture, we will present what are zoonotic diseases, some types, the transmission. We will continue to talk about ways to prevent zoonotic diseases, some challenges in controlling it, and end, of course, with a summary. Now, what are zoonotic diseases? So these are diseases transmitted from animals to humans, caused by bacteria, viruses, parasites, or fungi. Why is this important? Because over 60% of human infectious diseases are actually zoonotic. Now let's talk about some types of zoonotic diseases. It will be based on the cause. So is it viral, like rabies, the Nipah virus, or the influenza? Or is it bacterial, anthrax, Lyme disease, leptospirosis? Is the cause parasitic, like toxoplasmosis, tapeworm infections? Is it fungal, due to ringworm? And another way to look at it is, what is it based on in the form of transmission, or what kind of transmission is it based on? So either direct, so it's in contact with the infected animals, like in rabies, or is it indirect, like vectors, like mosquitoes or ticks? For example, mosquitoes spreading malaria and ticks spreading Lyme disease. So two ways to differentiate. One is based on the cause, and one is based on transmission. Now, transmission of zoonotic diseases. So what are some routes? We have direct contact, like we just mentioned, so bites or scratches or handling of animals. We have foodborne, so eating contaminated meat or dairy, for example, catching salmonella. Waterborne, drinking contaminated water like leptospirosis. Or vector-borne, carried by ticks or mosquitoes or fleas, etc. Now let's talk about some examples of zoonotic diseases. We mentioned already rabies, which was viral. So it's a viral disease spread by bites from infected animals. So in this response here, you can already identify the cause, it's viral, and the way it's transmitted, direct contact, bite. Lyme disease caused by bacteria transmitted by ticks. Zika virus spread by mosquitoes, affects pregnant women and fetuses. Now, what is the role of the wildlife in zoonotic diseases? Well, wild animals are reservoirs. So bats, for example, carry coronaviruses and rabies. Rodents spread hantavirus and plague. Ecological changes, so deforestation and urbanization, increase human-wildlife contact. Now to some impacts of zoonotic diseases. Health impacts include high mortality rates. For example, in Ebola, long-term complications like birth defects from the Zika virus. Economic impacts, healthcare costs, trade restrictions, loss of livestock, etc. And social impacts, fear, stigma, misinformation during outbreaks. So zoonotic diseases, they don't just affect our health, but they actually disrupt economies, create fear, spread misinformation, especially during pandemics. Now let's mention the One Health approach. And it is a collaborative effort to achieve optimal health for people, animals, and the environment. Key principle here includes recognizing the interconnection of human and animal health. And the idea is to indeed have a collaboration, an interdisciplinary collaboration between medicine, the vet sciences, and environmental science. Example of this would be to vaccinate dogs to prevent rabies in humans. Now, preventing zoonotic diseases. So some strategies include vaccinating animals, like we just mentioned, for rabies, practicing safe food handling, using protective measures like mosquito nets and gloves, reducing wildlife contact through habitat preservation. What are some challenges in controlling zoonotic diseases? Well, there are barriers like lack of surveillance in low resource settings, climate change increasing vector habitats, globalization spreading diseases faster. Solutions: strengthening global surveillance, invest in research for vaccines and treatment. What are some emerging zoonotic threats? Example, the H5N1 avian influenza, spreading from birds to humans. The monkeypox, rising cases due to increased human-animal contact. The Nipah virus, spread from bats through contaminated fruit. Focus areas: strengthening early warning systems and collaborative global responses. And some success stories in zoonotic disease control. Examples include rabies elimination in many countries through pet vaccination, eradication of rinderpest, a cattle disease, through coordinated efforts, improved control of avian flu with better monitoring and culling practices. So in summary then, zoonotic diseases highlight the connection between human and animal health. Prevention requires collaboration through the One Health approach. Early detection and global efforts are crucial to controlling these diseases. And that's the end of this lecture series. Hope you enjoyed it and learned a lot. Thank you again.